Greetings, I'm Salvador Cordova. Welcome to the Evidence and Reasons for the Christian Faith video channel. And I'd like to extend special greetings to those who joined us, who just finished watching Dr. Michael Behe's interview on the Creation Myths channel, hosted by Dr. Daniel Stern Cardinale. And I have sent out invitations, private invitations to a discussion panel because open mics have been attacked recently on creationist channels. I'm not publishing the StreamYard link anymore and letting anyone come through. This is really unfortunate. I do enjoy talking to people. So let me see. Yeah, Dr. Dan. Yeah, he does talk, but I guess it's his show, so he can do that. If I tend to be more to ask questions, and he does talk, but I guess it's his show. So hang on, Angelo, we're getting some feedback here. I'm going to have to mute. If you can mute yourself.
Yes, I just did. I muted.
Okay, great. Thanks for joining us, Angelo. So I thought it was an excellent exchange, and Dr. Dan is obviously very knowledgeable about the Lenski experiment. He corrected Michael Behe on one point. I have my own views about the use of irreducible complexity in arguments. I thought that some of the challenges in some of the challenges of presenting the ID argument were kind of highlighted here. Dr. Dan was asking repeatedly, how do you draw the line? I wanted to point out that, and I did point out in the chat, the problem is evolutionists don't draw the line. They'll take one measly example of maybe some positive mutation and extrapolate that, and that's no proof. And so I just found it ironic that there was a demand to draw the line for irreducible complexity. And I just said, the problem is evolutionists don't draw a line. They accept everything on faith, even when it doesn't look plausible. And I found it also pretty ironic that Dr. Dan was accusing Dr. Behe of cherry picking when Dr. Dan's examples come almost exclusively, a lot of the ones that he likes. I shouldn't say exclusively, so forgive me, I didn't mean, I take that back. He draws a lot on viruses, and I've always challenged him. I said, viruses are a terrible model organism for the evolution of something like a human being. So those are some of my thoughts. Now, I just, in the spirit of full disclosure, I've not been a big fan of using irreducible complexity as a term, as a definition, and saying, well, this is a problem for evolution. I do not argue that. I will argue specific systems. I will argue specific systems and say the eukaryotic homologs that are found in prokaryotes, they can't evolve by small incremental steps. I can make that assertion, and we have plenty of evidence prokaryotes don't become eukaryotes. We have to appeal to exceptional events that we can't see, and that's what I found problematic in evolution. So Otangelo, go ahead and share your thoughts, and thank you for joining. I don't know how long this will go because this is an afternoon show, but I'm glad you were able to join.
Yeah, I mean, I don't very well know the thing which Dr. Dan talks about the virus and so forth. I had a conversation with him on irreducible complexity. He came up with the same two examples which he came up today with Dr. Behe. One was the HIV and the other the placenta. And since I was not familiar in my debate with him about these two, I had to say, well, I look it up, and then eventually afterwards I can give an opinion on this. And then he said, but well, let's suppose that it is indeed irreducible, as I am saying. That would mean that the claim of irreducible complexity has been falsified. And I wasn't happy with that pointing this out, because I mean, I have collected so many different biological systems which evidently show that they are irreducibly complex. And then they said, well, in the way that you are saying it, it means that irreducible complexity becomes unfalsifiable anymore. If you have one example which is demonstrably evolving, a system which is irreducibly complex but came by evolution, then Behe's claim of irreducible complexity has been falsified, and then you need to make a new hypothesis. And to confess, I remained a little bit struck in that and moved a little bit away in regards of that. But I mean, it is obvious there are so many systems which we know are irreducibly complex. The cell itself, it has a minimal threshold, and when you go below, then the cell can't be alive anymore. If you remove just one helicase, it cannot self-replicate anymore. If you don't have topoisomerase, which you are so familiar with, then the DNA strands cannot be disentangled anymore. So this is obvious that the cell requires a minimal number of parts in order to be able to start to be a living thing. So, and yeah, and I mean, what do you do about the two informations? If on the one side, let's suppose that what Dr. Dan says is true, that the HIV example is irreducibly complex and did actually come by evolution. And on the other side, we have another irreducibly complex system like the cell, which we do not have any evidence whatsoever that it could have been the product by evolution. Even more in principle not, because evolution depends on DNA replication. So what do you make based on these two contradictory things? So maybe what you are saying is adequate. I mean, we talked about this previously, and you said, well, I don't like the term irreducibly complex because it applies to so many different systems. It's better that you pick just one and say, this could not have evolved. Your microphone is...
Sorry. Or you could pick several, and I don't use the word. You could say, this is problematic for evolution, here's why. This is problematic for evolution, here's why. And then I point to that. I think that's more successful. Throwing all these terms, it just adds unnecessary complication. And the other thing, I know that we're all ID advocates here. It's very hard. I wouldn't say very hard. It's easier to question evolution and critically than to actually put a positive case for ID. That may sound like hard medicine, but ID proponents are so eager to prove their point of a designer that they don't spend enough time pointing out all the problems in evolutionary theory. What I'd like to say is, look at James Tour's critique of abiogenesis. He didn't put forward the ID hypothesis, and therefore it didn't become a distraction. He was able, because he freed himself of having to advocate ID, he actually made a pretty devastating case against abiogenesis. So I'm of the opinion that if you prove evolution as faulty, people that are sympathetic to ID will get it.
Yeah, I think there are two approaches, and I think the approach of Dr. Tour is very powerful, where he has gone into the minutiae of the problematics of chemical evolution and demonstrated why it is such a big problem. And there are two approaches. One is eliminative induction, where we have two hypotheses. One is design, and the other is non-intelligent natural mechanisms. And when you can demonstrate successfully that non-intelligent natural mechanisms are not sufficient to explain the complexity which we see in nature, then by default, the design hypothesis is true, even if we don't know how exactly God did it. That is one successful approach in my understanding. But the other as well is where you make an entirely positive argument, where you say, like I am doing, I say that the cell is a factory full of machines and comes by instructional information. And such things we only know come by intelligence. So you have basically two approaches to make a case for creation. One is where you demonstrate that natural mechanisms are not sufficient, and the other where you make an entirely a positive case of abductive reasoning to the best explanation.
Well, I have, before I forget, let me share this embarrassing anecdote. Dr. Behe, and this was like over 20 years ago, maybe 20, just a few years after. This is the 25th anniversary of his book. And I was an undergraduate at George Mason University when he was doing a book tour. So this was not quite 25 years ago, maybe 22. And it's long enough ago I could say this story. I was going to take out a young lady for her birthday. And she always, that she was really looking forward to it. The day before I canceled, I said, Dr. Michael Behe's coming. I need to go to his lecture. She never forgave me for that.
Dr. Behe is more important than the lady.
She took it pretty personally. So,
did you never marry, Saul? Are you single or are you married? I never asked
married.
You,
she was definitely someone that I might have. She was very talented. She played.
You lost to marry because of Dr. Behe.
Dr. Behe. Yeah. Boy, she was livid. She was livid. Because I was taking her out on her birthday, and I said, "Oh, can we, Jean? Can we reschedule? Dr. B, he's going to be in town." All right, so I just had to.
You should have invited her to go listening to him together.
Oh my goodness! See, we're I should. That didn't occur to me. That totally didn't occur to me. So, yeah, that was a long time ago. So that's how that transpired.
You should have said to her, I appreciate so much that I invite you to come listen to Dr. Behe with me. It will be unforgettable.
Yeah. So, yeah. I've blown a few too. I mean, while we're on the topic, just as a side note, yeah, I've one time I took a girl out to dinner and I was giving her a hug goodbye, and I called her by the wrong name. I said, "Thanks for a lovely evening, Sarah." That wasn't her name. She punched me.
Wow.
So yeah. Yeah. So I have, anyway. Oh yeah, there are probably a couple dates where could have gotten killed. I took the young lady out to I was a new pilot. We went flying, and I was just coming. We were just 50 feet.
You're a pilot?
Yeah, I was in the aviation industry. My employer sent me off to flight school so I could learn how to fly. So if you ever see some of my shows that have pictures of me, there's a picture of me in a flight suit. I was in an aerobatic airplane, and
cool.
Yeah, we were 50 feet over the runway, and that's a pretty vulnerable time. The girl took an inappropriate move to hug me right when I tried to control the airplane. We landed. I was just like pale, just like we could have died.
Wow.
So I mean, thankfully it didn't disrupt the landing too bad, but that that was I got my composure back. I didn't scold her for what she did, but that's not the time to hug the pilot, when you're 50 feet over the runway and he's trying to land the airplane. So anyway, enough of those stories. I'm going to share something here that now I had mentioned this in my discussion with Dapper Dino, and this is a specific quote from an evolutionary biologist. Andreas Wagner said, "However, fitness is hard to define rigorously and even more difficult to measure. An examination of fitness and its robustness alone would thus not yield much insight into the opening questions. Instead, it is necessary to analyze on all levels of organization the systems that constitute an organism and that sustain life. I define such systems loosely as assemblies of parts that carry out well-defined biological functions." And this is from Andreas Wagner's book, Robustness, in 2005. Now, I pointed out, this is a paper I wrote. It was published by the Blythe Institute. It's an ID-friendly institute. They're a science institute, but they're ID-friendly. And it says, I pointed out, ironically, systems loosely as assemblies of parts that carry out well-defined biological functions sounds remarkably similar to Michael Behe's definition of irreducible complexity, a single system composed of several well-matched interacting parts that contribute to the basic function of the system. So kind of the same language is appearing in evolutionary biology, and I found it amazing that it was in the critique of fitness that they actually started adopting this Behe-like definition of function. And I pointed, it's not that the evolutionary biologists have I lost my train of thought. Sorry. The problem is evolutionary biologists themselves have found problems in the definition of fitness. And they're seeing that the idea of systems being defined by well-matched parts is a good way to look at biology rather than reproductive success. So whether we use irreducible, I suggested let's not use irreducible complexity as I don't like the term. It is fair to say that systems are composed of parts that have to fit well. So we can talk about.
They don't have to only to fit well, but in order to have a machine for a specific function, you require a minimal number of subunits and parts which contribute to the function of the higher system. And if you remove a single part, then you lose that higher function. And in the side chat, Nestlik said, well, oxygenic photosynthesis isn't irreducibly complex because the subparts, they can have a different function. And I said, well, then you should maybe go and look up what irreducibly complex means. He completely missed that what we have to explain and keep in mind is that we talk about a system for a specific function. And if you lose that specific function, then it loses the significance of the hypothesis. You cannot say, oh, because X of the subunit X performs a different function, therefore the system isn't irreducibly complex anymore. That's simply wrong. We have to keep in mind that the question is, if you have a system that performs a basic specific function, if you remove a part and it loses that function, and it couldn't have evolved by a stepwise manner, it is irreducibly complex. And there are so many different things that we can name. I mean, Dr. Behe has restricted himself to just a few examples, like the flagellum, like the blood clotting system, and a few other things. But I am listing many different systems which we can mention as irreducibly complex. Even rhodopsin, which is the center player in vision, the protein is irreducibly complex because in order to capture, to perform its function, you need to have both the retinal, which is the molecule embedded in the middle of rhodopsin, and then the opsin protein. And if you do not have both parts, then it loses its function. It cannot perform its function anymore. And I mean, there are so many examples which you can give, you know. So, I mean, that claim that because Dr. Dan has mentioned two systems which supposedly evolved and then became irreducible, because of that, the whole concept isn't valid anymore. I simply disagree with that. And I think that the two examples were provided, they have not demonstrated to be genuinely irreducible and that they came by evolutionary processes.
Well, as again, I felt that I know this may be painful for some of my ID colleagues to swallow, but I thought Dr. Dan's critique today kind of showed why we shouldn't try to we should reduce kind of the use of IC. If we talked about, you see, there was so much stuff being, half the show was just like talking about definitions and where it applied and when it didn't. Half the show was invested in that. I thought it would have been more effective if Behe came on and said, okay, here is the monarch butterfly. Here's the problem with it. Here is eukaryotic evolution. Here's a problem. And we just talk about systems from start to finish and not talk about definitions and when it doesn't apply, and that's an exception. It gave Dr. Dan a chance to talk about the viruporin pentamers and stuff. It's like I'm just giving some advice on how to prosecute the argument in cyberspace.
I think the problem is really, and I think that Dr. Dan pointed it out with his question, where is the threshold? And I mean, Dr. Behe, he doesn't deny that evolution occurs, but where do you put the limit, what evolution can do, and where you require intelligence to make a higher order or more complex structure? And I don't think that has been quantified. I mean, there are some papers which say that in order to have just two or three mutations require 84 million years and so forth, and that might be the threshold. I don't know.
If the audience sees me looking here, I have a second monitor, and it's great when Otangela is talking. It gives me a chance to try to respond to audience comments and send out emails and Google things so I could show. And
I think that, you can Google. Something which I thought was interesting, which Dr. Behe mentioned, was that in order to make the mousetrap, the single parts, also they need to be fabricated. And he excludes that point because, as he said, well, then we should go back even to the Big Bang. And it's good that we remain just to the core system and claim that to be irreducibly complex. But I have provided many examples where even the biosynthesis pathway to make a subunit of a higher complex system is irreducibly complex. And the prime example is chlorophyll B, used to capture photons in photosynthesis, where the biosynthesis pathway requires 17 enzyme steps, and 7 of these enzymes, they are used uniquely and exclusively in that biosynthesis process. So these enzymes, they do not have multiple functions. They have just that specific function in that biosynthesis pathway. And if you don't go through the entire pathway, you cannot have photosynthesis and chlorophyll. And if you don't have chlorophyll, you cannot capture light, and the entire process of making glucose through photosynthesis breaks down. So, and chlorophyll is just a tiny part. There are over 25 different protein complexes involved in oxygenic photosynthesis to make glucose. So, how do you explain that? By evolutionary processes? You can't.
And there are other ones like that. And you know what they'll often do is use these phylogenetic reconstructions. And I've argued that there are problems that they don't even think about just because they can build a phylogenetic reconstruction. That is, they take a protein and say, hey, look, it's in this creature and it's in that creature, therefore it evolved. And I think just devastating case against that is we could take the homologs of topoisomerase in bacteria versus the homologs in topoisomerase in eukaryotes. All those have, the ones in eukaryotes, if presumably the eukaryotes evolved from a prokaryote, and we can take all the other scenarios, it'll still have the same problem. Those homologs have to be tagged both at the RNA level and at the protein level to have import-export signals to go through the nuclear pore complex. So just because you could build a phylogenetic tree and that you could demonstrate similarity doesn't mean that the export signals, the nuclear export and localization signals, both at the mRNA level and at the protein level, can evolve just because you're able to build a phylogenetic tree. And Dr. Dan did appear on that show. He said, well, you need to, unless, can you do any statistics? I specifically, and I may show that the problem graphically, if I can dig up the slide. He said, you need to put natural selection there. I said, natural selection would select against it because the changes would be lethal. Or at the very least, it's not going to be able to be successful. Like if you started having nuclear pore complexes and you didn't have these reformatted topoisomerases, the creature's dead instantly. That's an argument why nuclear pore complexes won't evolve.
I think actually that the issue of irreducible complexity is much more severe, Saul, than many do appreciate. And I can give you an example. For example,
could we hold on one
sure.
Just a little bit? I wanted to answer in his image. This also relates to Benrex. And sorry to cut you off. If you could just hold that thought, because I'm backlogged with two. This is a fitness landscape, and Ben Rex was saying, could you show a graph where there are all these peaks? It can go in and out. The idea of a fitness peak is that the creature optimizes a trait. Let's say, see if I could zoom this a little bit better. Let's say the creature optimizes a trait here. But to get to a peak here, he actually has to go against selection, and then he might be able to climb. So there's intermediates. This is our, we don't even have to talk about irreducible complexity. Even in modeling of evolutionary ideas computationally, it's a known fact that sometimes to make an improvement, the intermediate steps would be selected against. And this is a problem in both evolutionary computation, and then it applies to the biology. So even without appealing to irreducible complexity, this is a known problem. This is a known problem. So, and so when I was describing this whole thing of evolving a nuclear pore complex being detrimental to the system unless you're able to change things simultaneously, selection, what this fitness peak shows is natural selection prevents evolution. They keep saying it helps evolution, the building of complexity. It actually does not. this is a good illustration that people who aren't ID proponents began to realize that natural selection is an agent that prevents evolution of complexity. And in fact, Michael Lynch, in his book, Origin of Genome Architecture, was saying that he was kind of saying the people who are ultra-Darwinians, he didn't use that term. I don't have the quote handy right now, so forgive me, Dr. Lynch, if I'm for only paraphrasing, but he said, these guys, these ultra-Darwin leaning people are no better than intelligent design advocates. And oh my goodness, Erica is here. Thank you for gracing us with your presence.
Oh, please. I'm happy to be here. Can you guys hear me okay?
Oh, yes. And you have a great voice. And I wish I could talk as fast as you. I just you could see Dr. Behe and I, we kind of stumble and we stutter. I do have a slight speech impediment, so I'm very envious of how articulate you are.
Hey, it's a double-edged sword there, Sal. I get a lot of people who comment on my videos and ask if I automatically speed them up, artificially speed them up because of how fast I talk. And the voice is a little high-pitched. It can be grating. I know my limits.
Well, I thought it was a very great interview, pretty hard-hitting.
I did too. Yeah, I
thought,
did too.
And I just like, ID proponents may not like seeing their hero kind of get grilled. And, I think a lot of creationists are just reflexively, we're going to defend everything Michael Behe said. And I'm not doing that today. And I don't know if you heard my earlier anecdote, how I canceled a birthday date with someone.
Yeah, I did hear that.
So I am a big fan of Michael Behe and his work, but it doesn't mean that I'm not, that we should, just because we're creationists or ID proponents, that we have to necessarily endorse every argument he makes verbatim. And so that was my point. So I thought it was a very good exchange, and ID proponents should watch it carefully. I know they may feel very hostile toward Dr. Dan, but it's like, well, if someone wants to make good arguments, this is what you have to face.
Yeah, I think that's what made it such a good conversation. Honestly, is that both of these guys, they've got incredible knowledge in their respective backgrounds, and they could have a conversation about things that maybe those of us like myself who are not kind of biochemically or genetically minded can kind of follow along with. They can dumb it down for the rest of us.
Can you hold? I feel bad now. I had a cue here of Ben Rex and in his image, but then I had to hold Otangelo hostage. I promised that he could finish his thought.
Yeah, no problem. I'll shut up. I'll shut up.
So after Otangelo, Erica, then you'll have the floor. Otangelo?
Yeah, no problem.
No, Erica, you can go on if you want to say something. Otherwise, I finish what I was going to say.
Okay, cool. Yeah, I want to, this is y'all's channel, this is Sal's channel. I certainly don't want to overtalk. And as Sal knows, having been on my channel, I can be a victim of that. Over talking, I monologue if someone doesn't cut me off. I thought it was a really good conversation. I said something in the side chat earlier that I think was I didn't mean for it to, text kind of can come across as being, I guess, more harsh than it needs to be. But I've been doing a lot of research lately on intelligent design in general, kind of as this proposed hypothesis, and because I'm planning on making a video of it, and I read The Devil in Dover, a bunch of different books on the Kitzmiller versus Dover trial, which was when ID was kind of first brought up as this opposing hypothesis that could be taught kind of alongside evolutionary theory. And one thing that I kind of noticed, because I was reading some of the court transcripts, is that I felt that intelligent design as a hypothesis, as it's defined, and as it's kind of proposed to be put into practice as this opposing theory, hasn't made much ground since 2005. And I was corrected in the side chat because I forget who said it, but someone was talking about some new stuff that's come about come out about the flagellum and how the proposed precursors don't work, or that there's, different kinds of arguments going on about that. But that was not my point. The point that I was trying to make is that intelligent design as its own standalone mechanism for the biodiversity of life that we see today hasn't changed since 2005, in that no new mechanisms have been proposed, or criteria for design, or ways to kind of identify it. And I think what Sal said earlier was very poignant. There's this whole aspect of ID where it's like, here's problems with evolution. And there's a time and place for saying, we have questions about evolution. These are, issues with things that we feel we don't have a current answer for, et cetera, et cetera. But I think that for a hypothesis to be considered critically and considered alongside other hypotheses or theories in science, they have to be able to stand on their own. So they need to have a mechanism, they need to have a model, they need to have criteria even for identifying what design would be. And that's what my point was in the side chat, is that from what I've, this is kind of fresh on my mind, which is why I felt okay in saying it, I don't think anything that Dr. Behe said contradicted that statement, that ID is the same as it was 15 years ago. Or what would it be? Yeah, 15 years ago.
25 years ago.
But yeah, well, since his book, but I was thinking the case. Yeah, 25 for the book, 15 for the case. So that's the only piece that I really had. I mean, I think that we could spend a decade talking about how, do we have an answer about how, X mechanism evolved? Do we have an answer about how X structure evolved? And I think that those are good questions to ask. But I think if for folks who want to assert that intelligent design is a viable alternative, it has to meet the same criteria that all other theories or hypotheses do, which it has to be empirical and testable and quantifiable. And I think Dr. Dan asked Dr. Behe a couple different times, like, okay, well, how can we quantify it? What does design look like? And Dr. Behe seemed to imply, if memory serves, I could be mischaracterizing him, and I don't mean to be if I am, but he seemed to be asserting that it's just intuitive. But different people's intuitions vary. So that's qualitative, not quantitative. Anyways, that's my 2 cents.
I think your criticisms are well placed, and I don't endear myself to other creationists, especially standing for truth when I say certain things. Well placed. I feel that if people ask me, do you think ID is science? I say no. I wouldn't classify it. My default answer is no. Why do you have a problem with that? There are a lot of things outside of science that are perfectly rational. William Lane Craig pointed out that there are many things we accept as rational and true that fall outside of science. And I actually have to go back to one of his debates where he articulated, he said, like, the only example, unfortunately, that I remember that he put forward is he said, it's perfectly rational to say that the Holocaust was a bad thing. That falls out of science, strictly out of science, but it could still be a true statement. And there were some other examples he had. So I wish that ID proponents were spending less time trying to say this is science and it can pass the test. You're just wasting time. I think the track that I use, and it's just one that resonates personally with me, is that I don't feel evolutionary theory, as far as, let's say we accept universal common ancestry as a given, even for the sake of argument. I have a problem with some of the transitions, and they seem like they're deal breakers with me. When I look at eukaryotic evolution, it looks like it needs miracles to make it feasible. And at that point, how is that really different than creationism? I mean, it's fair science to say we can make probability estimates based on what we know about chemistry and the way cells work, et cetera. I think that's fair. Criticism of evolutionary theory is scientific. I wouldn't go so far to say that ID is scientific. There is science that may support the final conclusion. So I will not endure myself. Some ID proponents were livid that I said that.
Right, yeah.
Well, I would like to answer to that, Saul. First of all, I think that we can make...
Tangela, can you hang on? I just want to welcome David Neff, and then I'm sorry, I have to cut you off. David, just greetings, and then I have to let Tangelo have the floor. So I didn't want to ignore you. Go
That's
on,
great.
Tangelo.
Yes,
Greetings, greetings to everyone. Thank you, Sal, so much for inviting me on. It's good to see you. Hello, Angelo. Good to see you.
yes,
And
hello,
Pastor
David.
Kevin, good to see you as well.
Absolutely.
Yeah, you're. By the way, you guys are the favorite evolutionists with me in his image.
We,
And
I,
Dr. Dan.
yeah, that sentiment resonates, Sal. I always reference you as my fave. I do. We do have a history though. We've been friends for a while, so maybe I'm biased. Okay, I'm sorry. I'll shut up. Tangelo, sorry. Go ahead.
Yeah, I think it is traditionally the main focus has been to criticize evolution as a viable option to explain common ancestry and biodiversity. But in order to be something more substantial, you also require to provide an alternative explanation, what actually replaces evolution. And I have summed it up upon my investigations, and I can quickly read it. The big umbrella contributor to explain organismal complexity and biodiversity, which falsifies and replaces unguided evolutionary mechanisms, is pre-programmed, prescribed instructional complex information, encoded through at least 33 variations of genetic codes and at least 41 epigenetic codes. Complex communication networks use signaling that act on a structural level in an integrated, interlocked fashion, which is pre-programmed to direct growth and development, respond to nutritional demands, environmental cues, control reproduction, homeostasis, metabolism, defense systems, and cell death. So that is basically my core explanation for biodiversity. And of course, in order to have pre-programmed information and information systems, you require intelligence.
So, can I ask a question, Tangelo?
Yes.
How do you identify what is pre-programmed, prescribed instructional complex information and what is not?
Okay, I think the best way to exemplify it is to go back to the origin of life. Okay, because there we require a minimal number of proteins, of molecular machines, which are necessary to have the basic functions to have a living cell. And I put that threshold, and I take as a parameter the smallest known free living bacteria, which is Pelagibacter ubique, and it has 1,350 proteins, more or less. Okay. Now, if we take that each protein has an average size of about 300 amino acids, and you do not have evolution as a driving force to explain the origin of life, and basically you have just to stick to random unguided prebiotic events, then the chance to have that minimal proteome is 1 to 10 to the 722,000 of power, which is in the realm, of course, of the absolutely impossible. But on top of that, and Dr. Tour brought that term up, and I investigated it a little bit as well, you require also an interactome. That means if you have these 1,350 proteins, you also require to have they networked in the right manner and to having an interactome where they can work in a joint venture together. And if you also calculate the odds to interlink these proteins in the right manner, then also the odds are 4 to the 4,000ths of power. So it is on top of the odds to have a functional proteome, you have also the odds to have a functional reactome. So yeah.
No, sorry to interrupt. The only thing that I would say about that though is like, so when you're coming up with a methodology or a mechanism, criteria is key. Like you have to have objective criteria so that you can categorize new data as it comes in. So I don't think that it's I don't think that that counts as a mechanism or as a model, because saying that you have an example of something that meets the criteria isn't the same thing as having criteria, because it doesn't act as something that you can then apply to new data. So you'd have to go to that free living bacteria and break it down and say these are the aspects specifically, you quantify it, so to speak. These are the aspects specifically of this free living bacteria that I think are, say, irreducibly complex. And here's why they are. So having an example, like, it's a good start, but I don't think that that constitutes a model or a mechanism still.
Well, I was replying to your question of how that information in the cell has to be pre-programmed. Okay?
Well, I was asking for an example of like how you differentiate the difference between. It's kind of like sorry to interrupt, but it's kind of like when I talk to creationists and they talk about created kinds, right? And this is at the macro level, which is typically what I'm referring to when I'm talking about evolution, because biochem is outside of my realm. I'm not a biochemist. But, you know, like, where does the evolution stop? Because many creationists, and I'm not trying to characterize you if this doesn't describe you, but many creationists are okay with certain levels of evolution within kinds. So my question is always, okay, where is the line? You know,
the problem, Erica, is deeper, because of course you can start when life already is there, but we do not propose intelligence as a mechanism only to explain evolution, but also everything, basically the entire created order, and that includes abiogenesis. And what we do know is that genes, they store assembly information, and that assembly information to make a device for a specific purpose is always tracked back to an intelligent source. And the sequence of nucleotides which direct the making of proteins, it is complex and specified. So you cannot have any kind of sequence. It has to be a specific complex sequence. And if you have not evolution to explain the origin of the first proteome, what is left if it is not intelligence? It is basically random stochastic prebiotic events which somehow sorted out which amino acids would have joined together to have functional proteins. And as I pointed out, by unguided events, the chance is far beyond what is possible, even if you shuffle during the entire lifetime of the entire universe.
So oh, sorry, go ahead, Sal.
I wanted to say, Otangelo used terms like specified and complex. I think that's been a failure in the ID movement. It's an ID they just need to get rid of. I'm sorry to say, you know, this may sound mean, Pete. That's why some creationists and ID proponents don't like me, because sometimes I'll say, you know what, you need to listen to your critics more because they actually have a good point. And just,
no, but you know, I mentioned as well.
And I wanted to say that, you know, we're too reflexive.
Can I chime in real quick?
Yeah, go ahead.
Sure. So, Sal, this is one of the reasons why you're definitely one of my favorite creationists, and because you are willing to listen to us and recognize the foundational flaws in some of the creationist arguments. I would say that my biggest problem, I would say, with just simple intelligent design is, okay, who or what is the designer, and by what mechanisms did this designer design life? Because you have to have some form of mechanism in scientific literature. You have to be able to say, okay, this is how this happened. This is who or what did it. There's thousands of different gods out there. Which one? Why? This is my biggest problem with intelligent design, I think.
Well, so it's the problem. I just want to say that's the problem if we're trying to make intelligent design a scientific argument or a mechanistic argument. I just briefly I said intelligent design doesn't have a mechanism in terms of a mechanical sense. What I mean by that is you can use quantum mechanics to describe the mechanical process of how certain chemicals form, and it's repeatable in the lab. We can't do that for an intelligence. You can invoke it, but it falls outside of science. So what I'm saying is, if we drop the requirement that it's scientific and repeatable, that we could say that, maybe the mechanism falls outside of any explanation, then that's fair. But to keep pretending that you actually have a mechanism that can be described like the other mechanisms in science, that just doesn't work. Okay, because I've worked with physics. You have very well-defined mechanisms of how things work. You can't do that with an intelligence. And so it's like, why do you keep, I just tell the ID proponents, why do you keep bashing your head against the wall and saying we have the mechanism? I'm just like, well, it may exist, but you can't describe it scientifically. It's very hard to describe what intelligence actually is. And so what we can say scientifically is something is improbable. And we could say that, as Dr. Cruz's been saying, it falls outside of maybe anything of our accepted laws of physics and chemistry. That's a fair statement.
Yeah, I agree with you on that critical point, Sal. Now, I would like to come back to your criticism in regards of using specified complexity. And I think for a layperson, it might be not so easy to understand what is meant by it. But I don't use just these terms. I also describe the information stored in DNA and in epigenetic information as instructional assembly information. Why? Because it instructs the making of the amino acid sequence. It instructs the sequence of amino acids, which is crucial in order to have functional folds, which will then become functional proteins. And that sequence is crucial. If you don't have the right sequence, then it's no deal. And coming back to irreducible complexity, and I was starting to say that I think that the problem of irreducible complexity is much bigger than many actually acknowledge and visualize. And I want to give an example. For example, all life forms require iron for cofactors which use iron sulfur as cofactors. And this is life essential. Now, to get iron into the cell isn't easy at all. There are specific assembly lines which transform iron in the environment. They chelate it into usable soluble form in order that that iron is able to be imported inside to the cell. And on the surface of the membrane, there are import channels. And these import channels or proteins, they have very specific, and it's just a few amino acid sequences which look out, and they are able to bind and recognize iron. Now, they have made knockout experiments, and they have given the result that if that sequence is not there, then the cell cannot recognize the iron on the surface, it cannot bind it, and it cannot import it. And that alone would simply make life impossible. So what does that mean? Yeah.
But there's no need to use the term specified complexity. There's no need. It's been a disaster. I was supposed to work on the specified complexity project as a graduate student. It's just as well that they shut down the lab before I had even any chance to go. I ended up going, instead of going to Baylor, I went to Johns Hopkins to study physics. I was actually going to work in the informatics lab. But now looking in retrospect, that whole idea has been somewhat of a minor disaster. No one can explain. It's just like, why use the term? All you have to do is just explain it the way you did. You don't have to add this superfluous term that no one really understands, defines, or can effectively use.
It's kind of vacuous, is it not, Sal?
Well, that's what I was trying to say. I'll give you a specific example. I asked,
let's just replace it with assembly instructions. And assembly instructions is a term that any one of us understands, and it describes perfectly what we have, the information that we have stored in genes. It is assembly instructions to make proteins. And assembly instructions to make something for a specific purpose is always the result of intelligent action.
Well, David Neff, did you have something to say? I just want to make sure you're represented here.
No, you guys can continue. Go right ahead.
There was some question about created kinds. Now, And then anything new in ID, I will tell you that there's an exciting project. Okay, it's exciting among ID proponents. Evolutionary biologists may not be interested in this, but let me see if I could. I did want to share something with you, Erica, because I don't know if some of the viewers here may have seen Erica and I actually do a
collab.
We did a show, a joint show, one of the few times creationists and evolutionists, we did a show on phylogenetic methods or building trees. Fun. So, Erica and I have had a friendly agreement that we're never going to debate each other because.
Oh yeah, we're friends.
Yeah, we're friends. I'll find someone else I can hate on, okay? But not Erica. In fact, that I remember my second debate with Dr. Dan. That's when it was your engagement was announced. So that was such a happy time. I said, yeah. And then James photoshopped you with your supposed fiance, and it was Kent Ovind.
Yes,
so that was me.
I keep telling my fiance we're gonna have the reception at Kent's Dinosaur Adventureland. Yeah.
Okay, so just on a more serious note, and this is something I try to tell creationists. You can't run away from these nested hierarchies. You build proteins, you will get nested hierarchies. So I'm a little bit concerned that we'll say that they don't exist and they're problematic, and then if someone becomes a biology student, he's going to do gene browsing and protein comparisons, and you can even automate it. It won't take him but 2 classes and he'll be able to build trees like this. And it's very evident, you could see here, that the human and chimp topoisomerase 2 are very, very similar. In fact, I've seen genes where the human and chimp are identical. And then we're able to build these trees out with other creatures, and it looks very similar to what evolutionary predictions would be. And what I was pointing out is that just because you can build a tree doesn't mean that they're not serious mechanistic problems. What I highlighted in red here is the topoisomerase. This is a nuclear, it's either an export or import signal. I don't remember which. I knew it when I made the slide. And that's the problem. If you don't put notes in your slide, you're going to forget what you.
Oh, I know that.
And now topoisomerase has both a nuclear import and export signal for the protein, but then it also has it at the mRNA level. And what I was saying is, and this is a life critical protein. For anything that has double-stranded DNA that needs untangling, it's a life critical protein. There are some like phages that are single-stranded, and I don't know how they sort of stay in a tangled state. But what this diagram shows is this is the nucleus, and then this is the membrane, the nuclear membrane, and then the cytoplasm. And I was just pointing out that if we don't have, even though we can build these nice little trees, something like this is some subtlety like this, like the nuclear localization or nuclear export signal, is missed by the phylogenetic tree. But it's really important, and I don't see that it's reasonable that this evolves in steps, because without it, it's not going to be able, the topoisomerase won't be able to transport to where it needs to go. And so this leads then to the question, why do we have these patterns of similarity and diversity? And so when you're asking for new arguments, this is my chance to advertise it.
Perfect.
So you're asking for the created kinds. And so let me show, this is a really quick protein exercise here, the crash course. This is the human zinc finger protein here. and it has a very obvious pattern with cysteines and histidines. That's pretty important. You can see it's non-random, really important for its function. And there are other subtleties here. Up here is the collagen protein, and you can see it also has a distinctive pattern. The glycines, the G's repeating every third pattern. That's also very, very important for its structural role. And you could see here that, so this talks about kind of created kinds. I'll say created kinds in terms of proteins. If we did like hidden Markov modeling and trying to unify, we'll find that the proteins really don't fit like a common ancestral model of one ancestral protein. And that's a discussion I've had with like Dr. Dan and others. They said, yeah, they'll accept maybe a universal common ancestor for the organism, but not for proteins. So this is one way you can actually visualize the problem. This shows that it's very hard to imagine macroevolution of some ancestor that became this or that, because it has very specialized. Now, the thing that's kind of cute is, let me see if I have it. I don't have it. Within species, across species, it'll make that beautiful phylogenetic tree. I don't have that slide handy. Within species, it'll make that phylogenetic tree, but the problem is you can't unify these. And let me see if this was the right slide. I was trying to make a point with all this. This may not be the correct slide.
Sal, you're on mute.
Sorry, I don't know how I did
I
that.
said I have to leave. You are already in good company, so see you
I was
next
worried
time.
my Wi-Fi conked out. I was like, wait a second.
So, thank you, Tangela, for joining. So I wanted to say that where I was headed with this is the question arises: why do we have these patterns of similarity and diversity? Why can we build these trees, these nested hierarchical trees? And the new answer, this is a creationist argument. I wouldn't necessarily say it's an ID argument. And there can be an argument to be said that the ID argument is actually very old and it's pretty much unchanging. So I'm not actually disagreeing with you on that. So what is new that I'm throwing out, and I mentioned this once after I think I had a debate after show, and I specifically invited Erica, David, and Dapper Dino, and I alluded to it then, and maybe I'll flesh out the idea a little more. So one thing that I said is I'm fine saying that ID doesn't have a definable mechanism. We could, except to say it's intelligence, which leads to the problem. Well, how do you define intelligence?
Right.
And so I said, you can avoid that problem if you just stop trying to say ID scientific.
Right. I think,
could just say, yeah.
Yeah, I was actually going to say something that I don't know if I've actually expressed this kind of on air before, but I think that as far as something that at least that attempts to make that attempts to make testable predictions, I think creationism as a whole does a better job at that than intelligent design, which is objectively more conceptual. Folks who are, for instance, young Earth creationists will make predictions that, the evidence that we should find as we move forward in, say, genetics or, genetics is a good example, should suggest that the majority of the human genome is functional, for example. Like that is a real prediction that has been made, or at least that has been suggested. I don't know if it's ever been proposed in like a peer-reviewed journal or anything like that, but it's certainly been suggested, which is a prediction. One thing that I would like to note with the topoisomerase slides that you were just showing is that I think for me, because my background, so I do primates. Primates are my forte. And something that I run into quite a bit is that young Earth creationists seem to have quite a large issue with humans being apes, great apes, hominids, and hominoids, and catarrhines, and haplorhines, etc. And when doing the Baraminology, separating out the kinds, they tend to do whatever can be done to separate humans as their own group outside of the rest of the hominids. And they, in my opinion, or not, I guess, I don't, this isn't even an opinion, this is something that I've seen quite objectively frequently, is that they will alter the criteria for separating kinds for humans only. They will use something standard for everything else, and then use something different for humans. And I see that as very problematic as far as it's not standardized, and as someone who has a scientific background, you know how important standardization is when you're categorizing things.
I wanted to echo that. And again, there are two kinds. There are creationists that fall in two camps, the Baraminologists and then the anti-evolutionists. I fall in the anti-evolutionist camp. I don't get along well with the Baraminologists.
Right.
And it's hard because some of them are my friends. But I will tell you that the camp that I fall into, and you know who I have worked for.
Sure.
These two camps wouldn't be on speaking terms at some of the conferences we've attended. So your criticism there, it's, I respect it. And I said, I wanted to tell the creationists, stop trying to emphasize the difference. We're very, very similar to the primates. It's unbelievable how similar we are. And I said, we're certainly more similar to other primates than we are to trees. It's like, it's not a great argument to say, oh, we're very, very different. I've said that if one wants to make a scientific type argument against common ancestry, it's really establishing that human life is recent. Because if let's say that the Earth is young, say hypothetically for the sake of argument that it's young, there will not be enough time for common descent. And so then I said, that'll settle it. I said, doing these genetical arguments is just going to get you into trouble.
Can I actually ask a question regarding that?
Hang on. I'm sorry if I could finish out a thought. Can I finish out? Because I didn't finish out my thought here.
Go right ahead.
Looking for my slides. And then, so after David, I'm going to get to Emery. So I could show here that this was the human zinc finger, and then the zinc finger in a pig. And you can build these phylogenetic trees. And that looks like common descent. Similarly, we could do that for collagen. This is human collagen, that's zebrafish collagen, and we can build phylogenetic trees for that. The point I was making, though, is you can't unify the proteins. And so this pattern of similarity, see if it's this one. I was arguing that this is a stairway to understanding. This progression is to help us. So we actually have model organisms that help us understand human biology. Now, the neat thing that's really advanced, if we do sequence comparisons, a lot of labs are doing this. They could just take, like, say, the sequences across a whole bunch of species that have homology for that protein. They can do structure prediction with it, which is really powerful because it's very hard to do structure analysis with X-ray crystallography. Even then, it's not always that good. If we can do that with just this, with just the sequences, that's great. One thing that I found, an argument that I'm actually working on, and God willing, some peer-reviewed papers are actually going to be coming out. They won't be creationist. They'll actually be bioinformatics, structural biology papers. If you remove the eukaryotes, you're not going to be able to resolve the protein structure. And I'm just like, there's nowhere that this should work unless there's intelligent design. And so I just threw that out there. And Emery, I'm going to try to get you on board. I'm going to send you an email. So David Neff, why don't you speak? I cut you off. And while I sent, this is what's nice about just having three people. We don't overtalk each other. So
yeah. So I have
go
a
ahead,
question
David.
about the 6,000 years you said about the hominid waiting time problem. I think you just cited. I'm not sure. I don't mean to mischaracterize you if that's but if say that is the case, then if you want to throw in like a global flood that wiped out everything but two within 4,300 years, there doesn't seem to be enough time to account for the biodiversity from there. And I do apologize if I'm mischaracterizing your.
I'll say that. Just let me say this: there would be problems. There are problems with the young Earth creation model, and SFT hates me for saying stuff like that. He said, "Go read some more YEC literature. Get up on it." Just like, dude, do you know who you're talking to? I'm a researcher in these areas.
Right.
If there weren't problems to solve, I'd have been out of a job. Okay, so
that's the beauty
anyway.
of science, is that we've all got unanswered questions. That's the fun part about being a scientist.
I'm typing a link here for Emery. I can't type and talk at the same time. So I think Erica, you're the kind that could do that. I can't.
Oh, no, it depends, honestly. Usually I just ramble slowly while I type one letter at a time.
So that, granting that there are problems with the YEC model, but let's just say, hypothetically assume that it's true and that somewhere the problems could be solved. What that would mean is I don't think there would be enough time for humans to evolve from, like, say, a bacteria to a human being. And we could probably extrapolate that and say that we can't evolve from a primate common ancestor. I'd say that's for young Earth creationists, that would be their go-to argument. The genetical arguments where we start doing gene sequencing, it's like, guys, I look at gene browsers a lot, and I've seen the pseudogenes we share. I've seen all sorts of proteins. I've seen proteins that are 100% identical. It's like, this isn't a great argument to make if you're like straining at gnats and letting camels through. Emery, welcome.
Hey, Sal, thanks for letting me hop in. Appreciate it.
Yeah, I mean, just for those out there, the creationist open mics have been getting bombed. And so I've had the invitation only discussions.
That's probably smart.
Yeah, I was in the one where John got bombed, and that was not pleasant. And it was split second, but it was unpleasant.
And I have, this is homeschooling moms are now starting to come to my channel. And it's like, I have to really, the conduct here has to be
Sal, I have no problem with you not making this public whatsoever. Like, that's why I specifically asked. Like, I'm like, I know that Sal's not going to put the link in the chat for me to hop in, so I'm going to have to ask for it.
Yeah, I think that's the safe bet for everyone, because YouTube can be pretty hardcore when it comes to those kinds of bombs. You can get a whole channel nuked because of it.
Yeah, I got bombed one day. I think it was from P-Mars who porn bombed me, and I had to shut down the entire stream and restart.
Yeah.
So yeah.
Wow.
It's no fun.
I've definitely been more careful about who I've been letting into my streams.
Yeah. Well, the thing is, like, you really got to be careful though, because the one that got into John's channel got in by pretending to be somebody who was actually active in the chat, and is somebody John knew.
Oh wow, yeah, that's
So you got to be hyper careful. Yeah. So, but yeah, but for once, that's something we can all agree on. We don't want our streams bombed.
Yeah, that's 100% backed on that. And I mean, the whole point of having these is open conversation, and I can't imagine the type of person that's like, I don't think that conversation should be had, therefore allow me to nuke the channel. Like, uncool, totally uncool.
Yeah. So the reason I hopped in, and I can't stick around for too long, but reason I hopped in is Erica, you brought up human primate baraminology, and that the people doing the baraminology programs are selectively changing the results. Is that what, I may be misunderstanding? I'm trying to remember exactly what you said because that was like 10 minutes ago at this point.
To summarize, I think that baraminology is unstandardized and inconsistent when classifying kinds in an effort to separate humans from the rest of the hominids. That would be my thesis statement of that.
Okay. Now that leads me to ask the following question: which baraminology method are you talking about?
Typically, the ones that I browse tend to be the stuff that comes out of the main organizations who kind of attempt to categorize organisms. Now, as far as how they're doing it, I've seen a couple of different methods. The most recent one that I've looked into was it was barcoding, and Raw Matt was the one who was kind of parroting it, but I don't know who he got it from. My inkling is that since he doesn't do a lot of genetics, he probably got it from somewhere. I reached out and asked. Oh, sorry.
Sal, can I share screen real quick? Because I think I want to show Erica something and see if this is what she's talking about. Just let me get the thing I want to share real fast. I have like 50 million files on my computer. I
know how that goes. Here, I'm also putting in the side chat the link for the mammal arc kinds, the baraminology that Answers in Genesis did.
Okay, I have seen that. Okay, I know what you're talking about.
Yeah.
Thank you.
No problem.
Thank you.
Yeah, and another great.
Sorry, go ahead, David.
Yeah,
I'm sorry.
Another great example of this happening is the notorious Tompkins paper that allegedly showed humans having only 75% genetic match.
His revised, to be fair, his revised was 82, I think, after he did used the debugged version. But the problem with that is he did not actually weight any of his DNA kind of highlighted regions, I guess you would say. So Tompkins' method would actually end up separating individual humans by a greater degree than 99.9, for instance. But he doesn't touch on that, which is very strange.
Of course. And it's interesting that Answers in Genesis has not retracted his original paper. Because if I were to publish something like that in, say, Nature or Science, and it was discovered that there was a significant computer bug that completely messed up my results, this would warrant a complete retraction from the journal. And they also don't seem to have any link to the new paper on his old paper. So that should have been completely retracted.
I'm not going to comment on that because I don't know anything about that. Erica, is this what you were talking about?
Let's see. Yes, but I believe this is on dinosaurs. Yes.
Right, that one's on dinosaurs. I just pulled up an example of that particular type.
Yes, this is the type of methodology.
So I can stop sharing now. We're good. All right, so that's what I thought you were talking about because I know where he got that. He was introduced to that through Dr. Matt McLean and the thing that came. Actually, okay, so he may have gotten that through me accidentally. So let me kind of give you the background here. I had Dr. Matt McLean on to talk about baraminology, feathered dinosaurs, and the stuff that he did for his 2018 white ICC paper, which I vehemently disagree with. But I had him on to talk about it. And then Standing watched the video, and Standing said, I want him to come on and talk about the mammal-like reptiles, and I can't remember the technical term for them. I know what it is. Yes, thank you.
Yeah, no problem.
So Standing then had him on to talk about synapsids, which is where I think Raw Matt got the baraminology stuff. So that's what's going on with that. I have some serious objections to the way that particular baraminology program runs, and I'm working through that on the blog right now. But that particular program, I will agree with you. It's not a very, it's not the best way of doing baraminology in my personal opinion.
So oh, sorry. Go ahead, Sal.
I just wanted to say there's also been a minor fight as to how baraminology should be conducted. The two founders had a bitter dispute, and I had a discussion on biotic message, one of the founders with Walter Remine. So Go on. I just wanted to point out that's another problem. There's not unity on how to define baraminology.
There sort of is now, Sal, because basically the people who were on Remind's camp have either stopped working in the field or died off. Not to be mean, but that's kind of where it is now. Now I'm trying to work on reversing that because I think Remind was right and I think Wise was wrong, but
God bless you.
You may be cursing me. I'm not entirely sure, depending on how this goes.
Yeah. Erica, I'm sorry I cut you off.
No. The only thing I was going to say is that I think that's I totally agree with Emery on that one. I mean, because that's my problem with what I have seen of baraminology is that what I'm asking for is, okay, let's get something standardized. Like if we're going to look at these created kinds and take them at face value, which is what I always think should be done. I think anytime a hypothesis is put forward, it should be taken at face value and evaluated on its own merit. Because that's how good science is done. But my problem has always been what I've seen, and admittedly, I'm certainly not as well versed in baraminology as probably you are, Emery. But from what I've seen from AIG and the barcoding stuff, is that it doesn't tend to have that standardization that can be universally applied. And from what I understand, and I think I'm quite decently informed on kind of young Earth creationism in general. I think I am. Maybe others would disagree, but humans being hominids and having evolved from previous primates, of course, is kind of a no-no. And so I struggle because, as Sal mentioned earlier, it is not it really can't be disputed how similar we are to the other hominids. And so I'm always wondering, okay, well, how's it going to be done? How can we come to the conclusion that humans aren't other apes, and also consistently create kinds that are going to be reasonable for, say, the Noachian deluge, right? Because if every species is a kind, you reach a problem of fitting everybody onto the ark, which is why kinds were kind of, I guess, fleshed out more in the first place. One of the reasons, I suppose, kinds were fleshed out more in the first place. But being someone who looks at primates, when I look at what Answers in Genesis has put out with the mammalian ark kinds, I find it very interesting how they've separated out the primate order, because you've got just an absolutely bonkers range with the Cercopithecids, from talapoins all the way up to the biggest monkeys, the biggest ground monkeys being mandrills. And you've got way more diversity in that group than, say, the pongids is what they're referring to them, but it's hominids sans humans. And so I look at that and I'm like, okay, how is this being done? And that tends to be my issue with created kinds, because being someone who is hopefully, if my thesis gets approved here in the next couple months, a primatologist, right? We'll see. Fingers crossed. I'm getting my first. I sent my first thesis draft off last week to be edited by my advisor. And when I sent it to him, I was like, I'm ready for you to edit this. And then in parentheses, I put eviscerate, because that's what he's totally going to destroy it. And that's what I want. That's good science, I guess. But, that's just my 2 cents on that. And I tend to get criticism from some of the young Earth creationists out there, because I keep things pretty narrow in scope when I'm talking about evolution, because that's what my specialty is. And people don't seem to like it when I say, look, I'm not an expert in this thing, but why would I talk about something that I'm not an expert in when you could just talk to someone who is? Anyway, 2 cents.
Sal wants to hop in, so go ahead, Sal.
I just wanted to say, for some of the viewers, they may not realize what's going on here. Erica, you are a graduate student in primatology, biology-related field. Emery is a creationist. He's a graduate student in biology. I consider myself a graduate student in biology.
Sal, I don't think you ever stopped.
We have David Neff, who's going to be a biology student. What's going on here is people like me, Erica, and Emery might be in the same school, and we have to. It's natural for us to cooperate, and we may have conversations like we're having today among fellow peers. This is why this doesn't look like your typical internet screaming session, because both Emery and I have had to matriculate through secular grad schools, secular undergrad and grad schools. You have to learn how to cooperate and be helped by your peers. I had professors that were anti-creationists that helped me through school. So if people wonder why we're so collegial here, it's because we're colleagues. Yeah. Just wanted to throw that out for those who may not understand what's going on here. So this is this. If this sounds like a conversation among grad students, that's exactly what it is.
Absolutely, yeah. And I mean,
or
that's
as I colloquially refer to it, a nerd session.
yeah. Well, hey, listen, we've gained viewers since we started nerding out. So I'm just saying, it's
Hey,
people. There's a market for it.
Apparently. And just to kind of back Sal's point up, like I have had at least one professor who I'm pretty sure was I didn't ask him, but I'm pretty sure he's very of an anti-creationist. And there's a good possibility that I will be using him as my mentor for a research project.
Yeah.
So
isn't it nice when you to a lesser degree, but I have had one of my advisors, I guess. I've got my supervisor and a team sort of advisors that I've been bopping my pieces around to. But there's aspects within the narrow field of primatology, obviously, that there's much drama in, because every field of science has its own drama, things people disagree with. And one of my advisors had an opinion on this thing called the social brain hypothesis, which I just absolutely loathe. I don't think it works at all. And we get along just fine when we're not talking about that. So it's like you can always learn something from advisors who've been in the game longer than you have, even if you disagree. And that's the beauty of collaboration, right? Is that it's like you don't have to agree on everything as long as you agree on the subject of the research project.
Yeah, right. And just to kind of go back to where we were with the Baraminology project, I agree that a lot of it is kind of subjective, and that's kind of something I'm wanting to work on fixing. And I would make no bones about saying that I don't think humans are primates in the sense that we evolved from primates. Now, obviously I think we should probably the most similar to primates among all the animals, but I don't take that position based on I guess as a creationist, I'm required to take that position based on what the Bible says. And so I understand from the AIG perspective why they chose to separate humans out from. I believe you said the pongans. I'm not a huge primate nerd. Sorry, Erica.
No, it's okay. I forgive you, but reluctantly.
If I could throw this out,
Good.
It says in Ecclesiastes, "Men are but beasts," and
fair enough.
there's an aspect about us that's like every other biological organism. And
well,
we could say, okay, so where would you classify us? Would you classify us as mammals? I think that's
right.
fair. And so if we go up a little farther up, it's like is primate a classification just in terms of the architecture? We don't have to say common ancestry.
Well, and if I may, Sal, I'm so sorry to interrupt you, but I want to take this opportunity to agree with that point, because I've had conversations with creationists before, and they'll say, oh, you think humans are primates, and I'm like, well, yes, but if I wasn't a creationist, I would still, or wasn't a creationist. If I wasn't,
you may have just let the cat out of the bag there, Erica.
Oh, I know. Yeah, I'm actually a sleeper. I'm a real sleeper cell over here. If I wasn't a person who is into evolution, right, I would still think that. And there are many folks out there in the creationist community who do accept that humans are primates and catarrhines, and moving down that kind of lineage. Creation.com has an article about that as well, where they're like, look, we're looking at the genetics, we're looking at the morphology, aka how we define what organisms are, where they fall into the categories. And that's kind of where humans land. You could take humans out of it and categorize primates, and going down that line from primates, haplorhines, catarrhines, etc., and not include humans. You get the same organisms in the same spot, and then when you assess humans, that's just where they fall. So you don't have to accept evolution or be into evolution to look at the traits humans have and consider them to be primates and hominids, etc. In fact, even our mutual friend Standing for Truth has taken that position. He's like, well, humans are, hominids, I guess, like by the architecture. He's not super pleased about it, but that doesn't mean we evolved from them. And I would say that's true. That's a completely separate issue that we could certainly have a conversation about. But humans being primates and hominids, etc., has nothing to do necessarily with evolution, I would posit.
I think Linnaeus, who is a creationist, actually was among the first to classify humans.
You're correct.
It's actually interesting because Carlo Linnaeus, not only did he classify us as apes, when he also classified orangutans and others as Homo, as humans, he put them in the genus Homo, which was quite interesting.
Yeah, no, that was kind of that's a funny thing, because if memory serves, when Wallace was actually out in Malaysia, I believe it was, he encountered orangutans, and the locals called them the men of the forest, because they were just these kind of humanish looking things hanging out in the trees, and they were like, well, I guess they're just really hairy dudes, you know.
Anyway, I gotta run. Thank you so much, Al, for having me on. I really appreciate it.
It's rare that we are on the same channel these days, and different variety of reasons for that. But I wanted to extend hospitality to you.
Oh, of course.
You and Erica are my favorite evolutionists. So
We love you
Dr.
too,
Dan.
Cal.
Yeah, we love you too. You're
And
my
Emery,
favorite
I
creationist.
guess.
Yeah, you're my favorite creationist, Sal.
Oh, thanks.
And Erica, you're my favorite gibbon.
Thanks, David.
Anyway, have a good night, guys. I'm out of here.
Take care, David.
I think too. I know. Do you know Walker? Do either of you guys know Walker, Sal and Emery?
Yes, just a walking fish.
Yeah, he is hoping to, if you would allow him, hop in here in a little bit.
Yeah, let me send.
I think he's coming back from class, so I think it might be a couple minutes. But he wanted to hop in and chat if it was okay with you.
Yeah, I'm gonna do that. I especially like talking to students, fellow students, and it's certainly been. Oh boy, Erica? Can you talk? Because I can't talk while.
Oh, I totally
Try
can.
to talk and type again.
Man, being a student is a special kind of struggle, because you're in that. I'm sure you guys have felt the same thing, but there's that perfect imposter syndrome when you're trying to write up a thesis, where you're like, okay, so I'm doing this thing, I've run these numbers, I've done this stats analysis, and now I've got this paper staring back at me, but it feels like I don't know what I'm doing at all. Like,
Yes.
what even is this? Have I done anything right? And that's at least where I am right now, and going through. Oh,
Totally
sorry.
feel you.
Yep.
I look at papers that I'm about to turn in and go, do I even know what I'm talking about? Like,
Yep.
do I have any idea? And the thing is, I'll get them back and they'll have a pretty good grade on them usually. And I'm like, now, why did I, like, what was wrong with me?
Right. I find myself second guessing even like the basics all the time when I'm writing papers. I'll be like, oh my gosh, like this is just garbage. It's a terrible paper. This was when my program was kind of in person and we were doing like regular write-ups. Because we did a write-up on cortisol, the stress hormone in squirrel monkeys. So Cymeri, scurriensis, and things like that. Scurrius, sorry. And we wrote it up, and myself and a couple of my colleagues were sitting there. We were doing our write-ups because we'd run a lab and done all this stuff. We were trying to figure out if there was a relationship between a monkey's rank in its hierarchy and how much stress was in its urine. Because the hypothesis was like, okay, if you're lower on the rung, you're probably more stressed out. And we're writing it up and we're looking at it and we're like, this is trash. We're all going to flunk out. Like, they're just going to boot us from this program when we submit these papers. And we were like, well, what are you going to do? We submitted them. We went out to the pub because this is when I was in the UK. And then when we got them back, they were like, fine. Like, they was like, good grades, like not perfect or anything, but they were like, hey, this is a good write-up. We were like, how is this possible that we that we knew what we were doing? Is it possible we're learning? Are we learning things? And I guess the answer was yes, because it all worked out. But golly, that is not an easy path to walk being a student.
I appreciate some of the stuff that Dr. Dan just put in the chat. Like, apparently it never goes away.
Yep. Yeah. That and like past notes too, because sometimes I like go back and I read some of my notes where I'm like, yes, like this is how you write like a good paper. And I'll read the notes and then I'll read what I applied those notes to, and then I'm like, oh, I see. I don't know how to write like 2 years ago. This is just the worst thing I've read in my life. Like, burn it now.
Uh-huh.
It's it's the learning curve, I guess. What are you gonna do?
Well-
that's some of it, I think. Yeah, it's just I reread recently, made the mistake of rereading a paper I submitted my senior year of college, like not
Oh my
of
gosh.
undergrad, and I went, oh my goodness, I can not only can I not write, I can't read. What on earth is this? How did this get an A?
I can't even imagine the bravery to go back and look at a college paper. I
was looking for a citation because I knew I'd cited it in that paper, and I was rereading. I'm like, oh, this is awful. How did this get a good grade? How did I pass the class? What happened? How did I get my degree?
Yep. 100%. That is the eternal struggle, right?
Mm-hmm. Yeah, I totally. Anyway, and even like just writing something up simple that like I just look at it and go, okay, because the field that I write in a lot is creation evolution, so I know I'm going to get a lot of scrutiny. So when I write stuff, I look at it and go, okay, did I say this in a way that even if someone is incredibly dense and really doesn't want to understand me, no matter what I'm actually saying, because there's people like that on both sides, will they still get it? No? Okay, how do I rewrite it so that they do?
Yep. I learned the hard way by submitting a early paper at the beginning of last year year before last for the master's program, that my language is too flowery. I'm too romantic with biology. Like I'll be like this incredible feature or whatever, and they'll be like, no, that is not how you write scientifically. This is not a novel.
No, romantic with biology means you love the field. It's sometimes hard to not use those words.
It is. It totally is. And you've got to be so objective, which I understand because it's supposed to be concise and very clear, and all that jazz. But man, I have to like curb myself. I'll go over back through it, and I'm like, no, you're betraying how much you love the subject here. Cut it out.
Yeah, scientific writing is required to be boring, dull, and dreary, and not allowed to be exciting at all.
Yep, exactly. Yeah, I think that's why so many scientists go out and they write like books, that for general consumption, because you're allowed to just kind of bleed all over the topic when you release it in kind of a book format. That's like, this is how much I love it.
Yeah, you're allowed to completely nerd out about the topic.
Oh yeah,
which you can't do in a science paper. I'm sorry, just like the reviewers would crucify you.
Yeah, you wouldn't. Yeah, absolutely, and it's such a bummer. There's nothing I love more than flowery language expressing how much I love. Well, in this case, monkeys. But I may be alone on that. I'm still coping from that stinging statement there, Emery, on not being a primate nerd. That's so tragic to me. But dinosaurs are pretty cool too. I will say that.
I'm honestly not as much. I love dinosaurs, but I'm not into the biology of dinosaurs so much. Although I do have some fossils, so that's cool. But my primary nerd outs are on fish and plants. So there really is like, name a biological organism. Name an organism. Someone is really into it somewhere.
Yeah, like I took entomology in undergrad, and my entomology professor was like, he was just such a gigantic fiend for mites. Like, he loved mites, and apparently he was like one of the forefront guys on mites. And I was like, that has got, like, imagine being a celeb in the mite field. Like, you're people are like, what do you study? And you're like, mites. Like, okay. But I respect it. I mean, apparently he's got a lot of cool stuff out there about mites, and stones and glass houses. I like monkeys, so.
Yeah, I mean, I hear you. Like, I'm getting ready to submit my application for my research program and with my master's, and I'm trying to narrow it down. Like, okay, what can I actually do in my apartment, because I have to do everything distance? What can I actually do? It's like, okay, There's a lot of stuff I want to do.
Oh, yeah.
What can I actually get away with in my tiny little apartment?
Yep. Oh, yeah.
That isn't going to get me, kicked out, evicted.
The landlord will boot you for experiments in your.
Oh, I had an experiment when I was 16 years old in my backyard that sent me to the hospital. Could have killed me.
Oh my God.
I was building a rocket engine, but it had a metal casing, and the pressure built up. It exploded. I still have a shrapnel wound here in my chest.
Oh my gosh, Sal.
If it didn't get caught in my chest, it probably would have gone in my heart, and I wouldn't be here. And how I didn't lose fingers.
That is dedication to the
for that.
subject.
So, Sal, like, I don't even have words for that. Like, I
get my people. People are like, you got yourself into some mischief. I said, yeah, that was age 16. My parents come home, and I just took a nap because I was bleeding so bad. It wasn't such a. And I went upstairs. Told Dad, I said, I can't take the trash out tonight. And he said, why? And I explained. He said, I'm taking you to the hospital right now. And then he tells my mom, and she said, I'm taking your son to the hospital. And she said, he didn't want to explain.
You didn't go right to you spent hours without going to the hospital?
Yeah.
How were you not dead?
That is hardcore, Sal.
Yeah. So I was there for I was in the hospital 2 days so they could do shrapnel extraction. And then, let's see, later in life, I got in a hang gliding accident and I thought I'd never play piano again. My arm was shattered.
Oh my gosh.
Yeah, I've had my share of mischief. And then after that, I got kicked out of casinos. I was one time detained in an Indian tribal casino, and you get released if the tribal elders release you, because they're not, this is an independent territory. They're supposedly not under the federal law. So
you've had some misadventures there, Sal.
Yeah, misadventures.
The amount of stuff that Sal has done and then somehow walked away from / gotten away with, it's like, Sal, you should be dead multiple times over.
Oh yes.
Sal, your guardian angel must be getting like a killer raise,
Yes.
A raise? You get a promotion and a raise. Good grief.
Yeah. Yep. I very recently I've been keeping trilobites in my garage. This was prior to when it got cold, so this was a couple months ago. But I grew some trilobites, and my idea was that I was gonna breed trilobites. Like I was like, this is gonna be really fun. Like or trilobites. I'm so
triops, right?
Triops.
I was gonna say, you've managed to breed trilobites?
That would be a totally thesis worthy.
Thesis? They should just hand you a professorship if you.
Yeah, that's Jurassic Park. Oh my gosh, I keep calling them trilobites. I do that in front of my fiance too, and he's like, "You mean triops, right?" And I'm like, "Okay, the fact that you're roasting me on this means that because they look like little trilobites sometimes. Like they just, they've got the longicaudatus, I think is the species, Pterops longicaudatus." But anyway, so I digress. I was getting in trouble with my fiance because he would walk out into the garage and he was like, "It smells like a fish market in here because there's so many triops. Like there's just too many. You've got to do something with these triops." And I was like, "Look, I'm running a breeding program out here. Okay, like you got to get on board." And he was just like, "I'm really excited for winter because they are not going to make it out here in the garage." And I was like, "That is a horrible thing to say." But they ended up, it turns out breeding triops is a lot harder than it looks. And also my last triops died before the next generation came to fruition. They're a little bit cannibalistic. So there was some breeding, but then there was some eating. So anyways, we'll try again.
Your fiance will love that.
Yeah, he'll love it.
I do some fish breeding on the side, so like I totally get that. But I think I have it a little bit easier because most of what I breed is live bearers, and that's like just throw them in a tank and give them the right water conditions, and they'll make more.
Oh, that is awesome. That seems like such a cool hobby.
It is. I've got 2 tanks with. I actually have a species that is either endangered or something along the lines that I'm breeding, which is I'm really excited about because they breed like rabbits. Like, I've got 2 full tanks of these guys, and I don't know what to do with them.
I totally believe, and that's the thing, because once you've got too many, like, what's the play? Like, how do you get them to stop? You don't. You just end up with a lot.
Yep, you do.
Okay, guys, I'm gonna head out because I gotta get some work done. I gotta be productive at some point.
Yeah,
but
I gotta be at a meeting in a couple minutes too, so I probably need to take off too.
Sal, if you keep this up, I know Walker wanted to show up. He said that if you were
Yes.
still going, he'd pop in, but I don't know when that's going to be. So,
Well, I could probably do a solo presentation, and someone requested he wanted to hear me play piano, and I do have a small recording, very brief one, just got a very impromptu. It's not the best, so I'm going to post that. But Eric, I keep wanting to call you Gutsick. Sorry.
I answer to both. I do answer to both.
Thank you. It was a pleasure. It's been a long time since we've talked, and I'm glad we're able to catch up. This is
Yeah, absolutely. This was very
great
enjoyable.
hanging out
Very
to,
fun.
yeah. And Emery, as always, thank you. So,
Thank you, Sal, for letting me hop
Yes.
in, and I'll talk to y'all later.
All right. See you guys. Bye.
Take care. Take care. So, Arpit asked if I have a, let's see, if I have a, post a video. This is not a professional video. The microphone wasn't all that good, but here you go. It's me with a practice session where I practice something very slow and then I speed it up. I think I played, okay, hang on. CRISPR-Cas, hang on. Let's see, let me post this. Okay. Let me post my public email here. I'm typing this. I can't talk and type at the same time. And okay, so just for those wondering why I've normally had open mics and just posted a link, the open mics have been recently bombed, attacked, and I just found out even David Neff's channel was attacked. So I did send out some emails. I'm sorry, I didn't mean to offend anyone if I left anyone out. Oh, and Dr. Dan, yes, of course. So I'm going to send Dr. Dan an invite here, and then I'll try to respond to CRISPR. Okay, so apologies to the audience here because I'm having to try to multitask, which I really can't do as far as talking and typing at the same time. So I just sent Dr. Dan a link, and I'd like to congratulate him on a great show. And I'm going to check my other email account. And I believe I'm going to get something from CRISPR here shortly. Dr. James Carter, I think, just told me he can't join, but thanks for the invite. And so, all right, so we're waiting Dr. Dan, and I just sent out an invitation to CRISPR. Let me just check on Dr. Okay. Dr. Carter said he can't join. Dr. Carter is a creationist. He's also a professor of organic and biochemistry. So, and Dr. Dan is welcome, CRISPR-Cas9.
Hello.
And thank you for joining.
Yeah, sure. I was just thinking that you, right as she was signing off, Eric said that she'll respond to Gutsick, and I have to tell you, there's I've noticed an uncomfortable amount of my head jerking to the side when I hear the word CRISPR said out loud, which, as you can imagine, in today's environment, is not necessarily uncommon in a biology lab. That's just kind of hard
yeah.
to explain.
So, and it's once you have a handle, it's very hard to get rid of it. So,
But no, well, I very intentionally created this profile, so this was a very intentional choice.
oh, well,
It was.
it's nice to finally meet you in
Yeah,
person.
I think we've
and,
talked in text a couple times.
oh yeah, a few times. And if you'd like to introduce yourself to the audience, that'd be great.
Sure. Go by CRISPR online. I'm currently studying in a PhD program for evolutionary biology. Previously done mostly cancer research for quite a number of years. So
Oh, thank you.
So
And-
broad background.
So you previously, I'm sorry, I missed what you said about you previously did cancer research.
Yes. Yeah, I was lab manager for a while.
What was your undergrad in?
Biology.
Okay. And Dr. Dan, welcome. Just want to congratulate you on an excellent show. It was pretty hard hitting. And I thought you had some pretty sharp criticisms. And some of the ones I've tried to,
I'm going to keep the
say
camera
I
off because I'm
proponents-
putzing around the kitchen making dinner.
Yeah. Okay. So just you go ahead and make dinner, but it's great having you on. And
I'm listening. I can hear you. I can talk. I'm not at the screen. I'm at the counter. I'm all over the place.
all righty, sounds good.
I don't know if you saw my advice relating to the bombing that's happening now, but that had actually been a problem a few years ago when people were still doing mostly Hangouts-based streams. And what we'd found was, first, if you put a join link in the description of a video, it's nearly 100% chance that somebody will join. Somebody's got an algorithm that is just searching video descriptions for join links, right? So, like, never do that. And then if you put it in the live chat, that's usually okay so long as you don't leave it there. So just somebody asks to join, post the link, delete the link, and you're fine. You know, you still want to like watch the person, right? But it'll cut down on most of it.
Yeah, what happened is I'm afraid we have actually some active stalkers too on top of it.
Okay, well that's yeah.
Yeah,
The
and I heard
verified
the name
accounts.
here that was pretty. Yeah, I heard something that was pretty disturbing just now. That someone who goes by the handle P Mars bombed David Neff's channel, and so he's going around in certain
I
in
gotta
certain
tell you,
communities, and that's just
it
I
is
said I need-
it's not a one-sided thing. It's attacking. It's vandalism is what it is.
Yeah,
It's not targeting one side or the other. It's just vandalism.
yeah, and this I mentioned this earlier that this is deliberately trying to be made into trying to keep this channel up to standards for family friendly, because homeschool mothers are coming here, and a woman in our church was recommending this website to 13-year-old girls. I'm just like, well, I'm flattered. It's not exactly, our audience is kind of at the PhD level, but we will have some material for the younger crowd at some point. We started at the very high scholarly level, but all this to say, that was kind of a wake-up call that the way that I have to conduct myself, and then also the way I expect guests to conduct themselves, we have to hold certain standards. Now, the nice thing is, since this is mostly a scholarly, nerdy channel, we usually don't get into fights. And
and
the language is good, but the only the risk now has been the bombing.
right, and that's actually a big part of the reason that I only said bombing and not the more descriptive title. Just to
Right. Thank you. Thank you very much, by the way. Yeah. But thank you for the advice. And the other thing is I may not actually be hosting very many live streams. It's very difficult because I have a situation where I'm the primary caregiver for my mom. She's going through a lot of health issues. She's 94 years old. She's going to be 95 soon. And there's a lot of intensive care. So it's only because Michael Behe was here today that I decided to have a live stream. So a lot of my stuff will be probably pre-uploaded, and then we'll have live chats. I'll participate. That's a lot less stress for me. I will join other people's streams because then I don't have to be the host, and I don't have to be operating all the controls and trying to control. That's multitasking while I'm trying to talk, which isn't good. Although I have to say it was where I used to fly around in the DC area. That was multitasking. You're trying to operate all the controls in the airplane, read the gauges, and try to talk to the air traffic controllers. It's like no wonder a lot of airlines have co-pilots and flight engineers in the crew, because you can have someone else talk to the tower while you fly the airplane, or vice versa. So,
It seems like one of those things where one person can do it, but two people prevents mistakes.
Yeah, it really helps. So that really was probably one of the harder challenges, because I do have a slight speech impediment, and so part of flying the airplane was actually just practicing how to talk and operate the controls at the same time. And anyway, that's neither here nor there. What do you think about the Dr. Michael Behe's interview? I thought it was Dr. Dan had a pretty hard-hitting interview there, but I thought it was technically accurate and fair.
Yeah, I think that, I mean, firstly, I'll say while he can hear it, what a fantastic get for Dan. So congratulations for that.
Yeah.
I think that it was very well managed. It's good for Behe as well that he's willing to go on to what he must have known would be a not aggressive, but certainly hostile show. I mean, and he did it, and I think both parties acquitted themselves nicely, and don't know what else to say. Obviously, I don't agree with Behe. If we've talked before, you know which side I'm on. So,
Yes.
But yeah, I think that it went quite well, and hopefully we can see more things like that in the future.
Yeah, this is, it's been hard sometimes because the ID movement and the creationist movement especially is very political, and they seem very oversensitive to criticizing their own. And I violate that axiom: don't criticize other ID proponents and other creationists publicly, and I do that a lot. And so my thoughts on the discussion, I've been trying to say, and I'm just repeating myself at this point, but for those who joined late, I said this is a good example of why I don't like trying to just concoct a term and then try to use it, because it's so, if you're trying to make a sweeping generalization like irreducible complexity, then you end up having to make all these exceptions. So I thought the first 20, 30 minutes of the show was just trying to define irreducible complexity and when it applies and when it doesn't to evolution. And as an ID proponent, I said, I would much rather just talk about systems of interest than these abstract concepts that have so many exceptions. It becomes just kind of unmanageable. So I thought Dr. Dan's exchange kind of bore that out, that this is one thing with actually trying to use the term rather than talking about systems. So what I thought was effective in Michael Behe's work, even though now it does seem to be a little bit more, in light of what we know now, some of the arguments are not as powerful anymore as maybe some of the more modern ones, I did appreciate his exploration of biochemical systems. And when I first read Behe's Black Box, it was so over my head when he was talking about the specifics. But that did give me a good introduction. Now, he did use a term that I liked that has actually been adopted by a Nobel Prize winner, kind of in a derogatory way. Behe used the term, it's a phrase that's known, Rube Goldberg. He was describing biology as Rube Goldberg, certain systems. I've seen a cell biology textbook use the same term. And then Nobel Prize winner Aziz Sankar used the term. He said the eukaryotic circadian rhythms are Rube Goldberg. But he used that in a derogatory way, but it's actually still accurate. And I've actually tried to say that that's a good way to describe biology. And people that know me know that I love showing small videos of Rube Goldberg machines, because it kind of conveys more of the concept of the way that I see biology as a Rube Goldberg machine. So those are my thoughts. I thought Dr. Dan had some really sharp moments there where he said, no, the promoter was not actually copied. The gene was copied beside the promoter. I'm like, yeah, that's, little minor points like that. And I was impressed with the fact that he'd read so much of what Behe actually wrote. I myself am not very familiar with the viruporins and some of the arguments of viruses for, as criticisms of irreducible complexity.
It's funny. I actually got that example, the HIV one, from Behe. That was a controversy. He alluded to it. It was a thing like maybe 10 or even 15 years ago. There was a big controversy. Yeah. It was the Abby Smith thing. Yeah. And it was that specific thing with the VPU and tether and antagonism. Exactly. So when I brought that one up, I specifically brought up Linsky citrate and HIV VPU as like, these are the two things that I know that you are really familiar with, because I've read a lot of your own stuff on these two things. So that was the specific reason I picked those two examples to like talk about, because it's not something like the I think my favorite example is the liposome transmission for the plasmids and the archaeal cells. But like if you haven't read that paper, nobody knows about that. So you can't just like, let's talk about this thing that only one of us knows about. That's not a good way to have like an interesting conversation for the audience, because one party is just going to be like, I don't know, tell me about it. But those examples I know that he was reasonably fluent in, so that's why I picked those. And CRISPR, thank you. I appreciate your feedback and your compliments. I'm realizing that kind of as I have, first of all, Sal, you remember we did a couple of debates last summer. It's funny, we can now say like last summer because it's been so long ago now. But and then I did a couple of other ones. But what I'm finding is, and I think people generally agree, is that a forum like this, where it's a less formal, less adversarial conversation, is a much better way to like actually get the points across for both participants, rather than feeling like you have to go tit for tat with like everything the other person says.
Yes. See, the debates are about persuading an audience in 1 or 2 hours. And
right.
Part of the problem is you have to educate them. And I'm just like, this is just, it's just begging for trouble. The only reason I do it at this point is if I get invited by, like, say, James Coons at Modern Day Debate, and if I feel it would lead to some advertising for my channel, and it's also good practice. But in general, I don't think that's.
Yeah, I mean, we've now chatted a couple times. I sat down with James Carter a couple weeks ago, Michael Behe just today, right? And that seems like a much just a better approach to like hashing this stuff out.
Yeah, well, some of my colleagues on the internet are like, I'm too friendly with the other side. I'm just like, dude, have you ever gone through secular school in biology to learn stuff or physics? One can't acquire the necessary knowledge. I mean, one might be able to do a program of self-study, but if one tries to matriculate through a secular program, you have to learn how to get along with other people and not just be getting in their face each and every time they say something you don't like.
Yes.
I'm just like,
Yeah, that's not going to work.
for all the times I'm sitting in school and my textbook and the professor says evolution or billions of years, it's like, am I going to stand up and make a fuss about it? Or am I going to try to learn and try to understand the arguments and also appreciate the criticisms that they may have? And this channel does appeal to biology students like Emery Moyna. And so it's like, yeah, they're not going to be impressed with these shouting matches.
Right. And
yeah.
the other thing is, it's funny because now, several conversations in and talking about different topics, and not all directly with people, but there was the exchange a while back with Paul Price and Rob Carter and John Sanford. They wrote the thing on CMI, and all of my critiques are kind of converging to just saying, show your work, please. And that's really all I'm asking for. Like, just show your work. If you're making a population genetics argument, show me the math on that. If you're making, like Behe, if you're saying there's a threshold, how do you calculate that? How can we evaluate that? Right? Just show me your work. And having a conversation like we did earlier is a better way to figure, like, to tease out those very in some cases it's the very fine distinctions that we're making, but they matter. And this, I think, is a better way to do it than shouting at each other.
Well, you and I are on the same page. To that end, before I forget, I did have a burning question. You said some of your creationist students approached you. I mean, if you feel free to talk a little bit about that. I'm just curious.
In general terms, I've had over the years, I've had a few students. One that I can remember in an evolution class specifically. So we chatted after class, a couple of times, and it was, kind of what you'd expect. Grew up in a, relatively conservative religious household and was always taught yada, yada, yada. And the nature of the conversation was, first, like, believe what you want to believe. That's like, doesn't affect me, doesn't affect this class. That's all you, and you shouldn't feel pressured or conflicted in any way with that. And then as far as the actual nuts and bolts of the material go, the thing is, you need to show me that you understand it. But if at the end of the day you walk away, if you could explain to me how, the different mechanisms for evolution work, but at the end of the day you walk away saying, here's how they work, thank you for my A, I don't believe that this actually happened. Like, I don't really care about that second part. I would like for you to come away thinking it's valid, but, like, people are going to believe what they're going to believe. And as an educator, my job is first and foremost to educate them on whatever the topic is of the class in question. And what I've tried to convey is that scientific questions can be evaluated scientifically, and we have a process for that. And then philosophical or religious questions, me speaking personally, are different, and you can evaluate those however you want. And I don't think that necessarily has a bearing on scientific questions.
Well, I appreciate what you said, and I had another secular professor who's anti-ID who had the same viewpoint, and that was James Treffel. So I really appreciate your treatment of the students, and I think the creationists in the audience I hope they'll respect how you treated that student. So what happened to me was James Treffel actually said some things that inspired. He wrote books on fine tuning, and he actually said, if I were a religious man, I'd say everything that we've learned in the last 20 years shows you're special in God's sight. That's not an exact quote. It's pretty close though. And I said, I think I want to study under him since he's at my school. My undergrad was at George Mason where he was teaching. And then I got an A in his class. I'd like to think I was one of his top students. And then some years later, he comes out and gives a big talk against ID. And so that was kind of interesting. And I stood up and I didn't get in his face. I said, Dr. Treffel, it was an honor being your student. And, I'm a creationist and I was your student. And he said, oh yeah, by the way, he got an A in my class. And so I raised the same question, what would you do? Do you have a problem with a student taking the exam and not believing what he was taught? He said, no. He said, I have to grade you you have to represent that you learned what I tried to teach in the class. But that's on you. And the reason that was important is I wanted the students, because, a lot of these Christians have a feeling of conscience that if they answer a question on an exam a certain way, they think that that's a profession of faith. And a lot of professors don't view it that way. It's just reflecting what you learned in class. It doesn't mean you have to confess it as truth. And so I appreciate you saying that. So I wanted to respond to something from one of the people in chat. The author of this paper won the Nobel Prize, but not related to specifically this paper. He won the Nobel Prize for something else in chemistry or physiology. But anyway, I don't know if you can see it here. It says the intelligent clock and the Rube Goldberg clock. And that title just kind of caught my, he was talking about the Rube Goldberg clocks in biology. And
Hey Sal, let me just jump in for a second because I am going to scoot because I'm about to be doing loud things in the kitchen, so I'm not going to be able to talk for a bit. So thank you, and thank you for having this and kind of bringing some viewers over to the Behe talk earlier. Have a good night.
You too, Dan. Take care. So I did send out invitations to some people in the private emails. If I missed someone who wanted to be on board, apologies. So this was Mr. Jetty was asking about something about Rube Goldberg, and that was something in. So that's an example of an actual peer-reviewed paper that described biology as a Rube Goldberg machine. And then I don't have the example of the cell biology text, but I'd seen one that uses the word Rube Goldberg to describe cell biology. And then there's this little article. It said cells as biological Rube Goldberg machines. Molecular pathways inside of cells can be visualized through and thought of as convoluted machines. And I know that gets under the skin of someone, especially because we call the cells machines. So if people wonder why we're still talking, I was waiting to see if Walking Fish would be able to join us. John Maddox, I did send out an invitation to you if you wanted to join the stream. So, CRISPR, did you have any other thoughts? I may not be able to wait around for Walking Fish. If not, I could, let me see. I think my mom looks okay for now. I could try to answer some of Ben Rex's questions. Speaking of which, Ben Rex 777, I've been very appreciative that you've been watching binging out on my videos. Just for the record, some of the stuff I've been doing in making videos, this is a precursor to fleshing out some ideas because I'm thinking about writing some small booklets. A whole book, I think, is too big a task. I think booklets are a little bit more. I don't think Walker could join. He mentioned he had quite a few things he needed to do first. Oh well, I sent him out an invitation anyway. So thanks for that. So Ben Rex, if you'd ever like to join these discussions, I can put you on my panel list. And so let me type you my email here. Like I said, I can't type and talk at the same time. If you'd ever like to join, now you did have some a lot of questions. Oh, okay. So walking fish. Thank you, Arpit, that he won't be able to join today, but he'd like to talk sometime down the road. So that's just an invitation, since you had so many questions and I couldn't quite get around to all of them. You did have some questions about James Tour stuff. And let's see how long. We've been going for 2 hours and 15 minutes. If there's any questions in the chat that I've missed and y'all would like me to address it, I'd be happy to. Let's see. I always enjoy a new source of information, especially if it isn't like most people on YouTube where you can watch a few tens of hours and then you know everything from them. Thank you for the comment. So there was a question, are Rube Goldberg machines alive? I'd say in general, no. But some of these, it's just like saying, is a cell in the human body alive? I'd say yes, but it's definitely not the whole human body. Are some of the processes in the cell alive? It's like, well, that's kind of hard to say. Like say a metabolic process, does it qualify as life? I wouldn't qualify it necessarily as life. I would qualify it as designed and for a purpose, and it's functional, and it is a Rube Goldberg machine. Now, the other reason that I like the idea of Rube Goldberg machine is it's obviously not meant to be a formal definition, but more of something intuitive. So in terms of Behe's book, which we celebrated the 25th anniversary, the Darwin's Black Box, he used the word irreducible complexity, and then the lesser known phrase Rube Goldberg, which was actually a phrase long before Michael Behe. But I think that's just qualitatively, I think that's a good description of some of the systems in biology. One Cow Stampede. If you'd ever like, let me just extend the invitation to One Cow Stampede. Typing here, you can join. I'm just going to tell them just email. So let me just post what I had written. Oh, yes. Thanks. For anyone watching, please hit that like button and comment. The other thing, I got 2 downvotes. So I wonder if that was the creationists not liking what I was saying. Anyway, can't please everyone. The Evidence and Reasons channel is the Evidence and Reasons for the Christian Faith channel. I also have a much smaller channel, about the 10th of the size, the Evidence Reasons Academy, and I may post a link to that, where I try to teach basic math, science, and chemistry to help creationists understand some of the terms. Now, I'm also doing this a lot for myself, that one way to relearn something or learn something is to teach it. So in the process of trying to teach these chemistry classes, I'm also kind of learning it too. So there's a question here. Do I need to download something for that? Hang on. It just moved. I'm posting the question. Do I need to download something for that? No. That's one of the advantages of using StreamYard, is you don't have to download software that runs on your system. You just click, and it does all the rest. It just does it straight through your browser, to the best of my knowledge, which is nice, because then it's not like Discord or Zoom that has to install something on your computer, which is really obnoxious. And so that's one of the nicer features of StreamYard. If I stated that wrong, anyone in the chat is welcome to point that out. So one reason I'm just requesting that you email me, you don't have to use a personal email address, use a public email address or a throwaway. It's just so that I can send out these links to people who I'm going to invite to talk on the channel. And same for One Cow Stampede. You've been a faithful supporter, and I appreciate that a lot. So, oh yeah, I was mentioning that I have this Evidence Reasons Academy channel, and I'm going to post a link to that. And over the coming year, I'm planning to have try to teach 2 semesters of college chemistry. Now, I actually think that if one wants to learn the ID and creationist arguments, one doesn't need to go through 3 years of, general chemistry, biology, cell biology, and organic chemistry, and then biochemistry. That's all well and fine if one is preparing to be a chemist or a biochemist or a biologist or a medical doctor or any of the other fields that would want a chemistry or biology degree. But to understand some of the basic ID arguments, I think maybe just setting aside maybe 20, 30 hours to really learn it could go a very long way. And you could spread that out over a year. I would even think 10 hours would help. If one thinks about it, James Tour's talks were a total of about less than maybe 10 hours. And yeah, some of it was pretty sophisticated, but I think some people were able to get the gist of it. So in the meantime, let me type another comment. My other channel is the ER Academy. Hey, my other channel is the Evidence Reasons Academy, where I will be giving lectures, God willing, on chemistry and molecular biology for free. I know that if an out-of-state student trying to take some of these classes, they'd be paying $7,000 at a regular university, out-of-state tuition, and I'm offering the class for free. That's the lectures. The reason that you pay more in school is they award diplomas, which I don't do. And they make you suffer through tests to qualify you. So with that, I don't have much else to say, but since Ben Rex is here, are you going to be here for a little bit longer, Ben Rex? You had a question about mixing entropy. And so now I'm going to put everyone to sleep. They can see now what some of my nerd stuff is. And I actually had let me close down some of my PowerPoints, and I'll bring up my mixing entropy PowerPoint. And this will be practice for me.
And just, I don't know if you saw in the internal, but I am back on to my mic, so.
Oh, great.
You can see the explanation of why, but yeah, I just had to take care of that. So
I totally. Oh, okay. I have to confess something. I almost never read the private chats because I'm so busy.
Okay, yeah, I didn't realize that. Yeah.
Oh, okay. Well, thanks for joining. Well, we're running out of things to talk about, and there is a viewer here, Ben Rex, who's been very faithful in supporting the channel, and he's been binge watching so much of what I've said, and so kind of in gratitude for that. And he did ask a question, and I promised, and I want to follow through with my promise. This is on mixing entropy. So this has nothing to do with Michael Behe. And let me see. Do I even? This is just mostly for the sake of one of the our loyal viewers here. And let me make sure that let's see. I have to go through this, make sure I have. Okay. I'm just blasting through my slides to make sure I have. Okay, because he had a question about, okay, this is going to relate to James Tour stuff. So sorry to disappoint anyone that, because I'm running out of stuff to say about Michael Behe. So how about this? It's going to be James Tour stuff. Some of you guys are okay with that, and I'll close this. I'll close this out. I'm going to close the after show out with James Tour because I think that's really cool. And I actually asked. We didn't have a question and answer with Dr. Dan because I know that's kind of hard to do, but I did pose a question. I said, I'd like to know if Dr. Behe watched the James Tour abiogenesis series.
I've not watched all of that yet. It's on my list, but
yeah, actually, let me see. Okay, so this is BenRec 777. And I think I could finish this in maybe 15 minutes. And this is kind of I'm going to give the gist of James Tour because he was very, very technical. And as I said, I felt his approach was more effective in actually communicating ID by avoiding the topic altogether. So that may seem strange, but I tried to say, just focus on the chemistry and improbability. And if people are leaning toward ID, they'll get your point. Don't have to try to sell people on intelligent design or creationism. Just talk about the science because that's a common language. That's just my approach. And James Tour did the job the way that I was very, very pleased with. So let me just close. Okay. So this is an unfinished draft PowerPoint of a presentation I was preparing to talk about mixing entropy. And it was partly part of the reason that I am interested in this topic is creationists have used the second law of thermodynamics to argue for creationism or intelligent design. And I said, please don't use it. That's not to say it doesn't have some relevance. There is one weakness in James Tour's talk that I kind of cringed at. He was citing Brian Miller's work on the second law of thermodynamics, and I'm going to cover that. And I actually had a little minor disagreement, both privately and publicly, with Brian Miller, who's a fellow. He's a director of some sort at the Discovery Institute. This is friendly. We're on friendly terms. We're on the same page. But I was trying to tell him there's no need to invoke the second law of thermodynamics. Go on.
I might have this number wrong, but doesn't life increase entropy at like 10,000 times the rate of anything else? I mean, it's just some outrageous number.
All right. Okay, this is great point. This is basic chemistry, and some people forget it. Water has a standard molar entropy. That means the more moles of water you have in a system, the more entropy that is. So what does that mean? And I pose this question: what has more entropy, a frozen dead rat or a warm living human adult being? What's the difference in entropy? And which has higher entropy? The answer is the warm human being has way more entropy, because entropy is what they call an extensive property. It is proportional to the number of molecules in the system. It's that simple.
But a few tons worth of frozen dead rats.
Exactly. The point is, okay, so just a single frozen dead. But the problem is, and you're totally right about a few tons, it becomes a useless argument. That's really my point. I'm trying to tell creationists, don't use that argument. You're going to get killed. Someone will just ask you a question like I did, or one that like you posed.
I mean, but the real point that I was trying to make is, so obviously, yes, you're certainly right that if you have a higher mass, you're changing the energy content of the system, and so obviously you've got more energy or more entropy then. But even if you have that closed system, right? And let's just talk about here we have a closed system, and it's starting with the same amount, the same energy content, same, right? And in one closed system with the same energy content, there is no life, and in the other one there is life. In which one does the entropy go up faster? The one with life goes up much faster. And I think, I think the number is something like 10,000 times. And yeah, so I don't understand this argument.
Even I haven't gone through that, and I couldn't answer your question. But I was just like, creationists, please just don't use arguments you can't defend or you don't understand. I mean, I specifically one of the topics I studied in grad school specifically because I was interested in this relationship of the second law of thermodynamics and intelligent design. I took statistical mechanics and thermodynamics, and after that class, I said, this is indefensible. You guys shouldn't use it. And then Brian Miller comes on and starts trying to use it. I'm just like, you can argue for problems in a closed system, but there are lots of open systems.
Right. To me, it's always seemed like if you want to use improbability arguments for abiogenesis, that's fine. But entropy, if anything, it's an argument. Entropy is an argument for abiogenesis, because if the goal of the universe is to increase entropy, and that seems like that's what always happens, right? Nothing increases entropy faster than life. So
yes, so my, that's exactly what you're pointing out is exactly why I want to steer clear of it. It just ends up being a mess, and people don't understand entropy. And even after I've studied statistical mechanics and thermodynamics, it's like, yeah, there's some subtleties that I'm still relearning and appreciating, and I'm having to ruminate because it's a very difficult graduate level topic. And even when one has gone through a graduate level program on it, it's like there's so many ways to mess this up, and it's just not worth going there when they're. And so that's about the only criticism I had of what I would call the major concern is like one of his opening shows talked about it, and I just kind of cringed at that point. So that's not a point we're in disagreement of. I wanted to highlight Zachary Sylvester's. He said, I'm going through Essential Cell Biology, Sal's recommendation. Yes, I wanted to point out, you can get Bruce Albert's Essential Cell Biology. You can get the first 80 or so pages for free as a free sample. If you have a PC, you can download the Kindle Reader, and then you can grab it and you can read it. And that's how you can decide if you want to buy the rest of the book. Now, you can get kind of free versions of it that aren't quite so good. And there is older versions of text that you can buy for like less than $10. They're like second edition paperback used. There is something at the NIH where they have his molecular cell biology, which is much harder, but you can get through pieces of that. So I am looking, and I want to acknowledge Dr. Dan, who got me started on this OpenStax and some of these textbooks that are free, that are public domain, and they do solicit donations at some point, and but their goal is to help students learn, and it's very commendable. And so I will look for an OpenStax like cell biology one at some point. So I wanted to salute Zachary Sylvester for looking through Essential Cell Biology. The reason this is helpful is sometimes just a few hours of study of this, you'll start to get familiar with all the terms that you're hearing. So CRISPR-Cas, so you teach students, is that correct?
Yeah.
Oh, so I didn't know there's VOET and VOET biochemistry.
Void, yeah.
I liked McKee and McKee, even though that wasn't the one I studied out of when I was taking biochemistry. I studied Leniger's principles, which was very technical.
Yeah, I guess I just that's I think that's probably one of the best upper division undergrad biochemistry books that you can get is the Void and Void. So that, I mean, there's matters of opinion there, but
well, no, thank you for the recommendation, and it really depends on what you need to know. I was told one biochemistry professor that I work with, he just laid out all these metabolic diagrams. He said, yeah, for some schools, they want you to just memorize all that. they'll test you on it. I was like, yeah, I used to know the citric acid cycle for a final exam, but I think a few days after I took the exam and, I recapitulated the entire diagram exactly, I started to forget it after 3 days. So let me see. There was one comment. Isn't Brian Miller's point that life has both low entropy but also high energy, and freezing rats come at the cost of energy? My main point is that whether Brian Miller is right or wrong, it's just making the argument way harder than it needs to be. This is thermodynamics in closed systems is hard enough. And then when we go into thermodynamics and non-equilibrium open systems, it just becomes a nightmare. And when I've actually heard Brian Miller argue against abiogenesis, he didn't hardly use any of the thermodynamics arguments at all. So I'm going to cover that a little bit here as I present it. And since CRISPR's here, he may be able to help me with some of when I fumble through the chemistry. So Tony Marie says pristine lab environments with the most purified chemical reagents and scientists still can't produce life. What I'm going to highlight here with mixing entropy, and that's the one aspect of thermodynamics that I'm kind of okay with, and I emphasize the word kind of okay with, is that it's not well understood why those pristine lab environments of purified chemicals is important. And it's even a little more complex than that. So that's what I'm going to try to cover for the sake of Ben Rex here, because he's been such a great fan of mine. So I'm going to share my screen, and I'll try to get through these slides as efficiently as I can. James Tour's 13-part series, which is actually 15 parts because he had part 0 and then 12.1, 12.2. It ended up really being 15 episodes that spanned about 10 hours. If I had to summarize what the main point he was trying to say for a non-chemist and a non-specialist like me, he used this picture of a Volkswagen that was disassembled. And one can see it has all these parts here. And so there's several levels with the problem of abiogenesis. At the very end, he said, okay, I'll give you all the parts. You have an assembly problem with abiogenesis there. And to a lesser degree, not to a lesser degree, but at another level of organization, even the parts have to be, the parts connect to each other. So one thing I wanted to point out is with like car parts, you can sort of tell that, the steering wheel doesn't really connect well to, like, say, a door. Okay. And so where this analogy fails is that in biology, a lot of the biological chemicals will allow you to connect very easily one part to a quote unquote undesirable part. And I'll explain what undesirable means, because that has a metaphysical implication, but it'll lead to pathways. Certain connections will be easily favored and preclude other kinds of connections.
You mean the covalent connections, not you mean the covalent bonds that might
yes,
form, not because obviously once you have these these biopolymers that have formed, they will like to group up with one another through non-covalent means, and that those tend to be pretty specific.
And they can be reversible.
Kind of like the steering wheel only wants to fit in with, wants to fit in with the steering column. These the polymers, they'll only link up in very specific ways once they've, on a sequence-based level. But I certainly agree that in terms of like your covalent sequence of building these molecules is a different question entirely. I just wanted to make sure that clarification was made.
Yes, and thank you for weighing in. And I'm by the way, you may have already answered my question, and I'm sorry if it went in one ear and out the other. You said you teach classes? Did you? You have students?
I'm a TA, so I lead a class, so I'm not a professor. Obviously, I'm still in the graduate program, but I am teaching.
Oh, that's wonderful. Is it chemistry related or?
No, it's intro bio type stuff.
Okay, so this is from my biochemistry, the Lenneger principles, and this is actually very providential. They actually happen to have this in their book. This is a nucleotide. If you could see my arrow here, my mouse cursor. This is a standard nucleotide here. But what James Tour is pointing out is that this phosphate group could easily attach to a place other than your standard. So rather than say right or wrong, it would be non-standard. Maybe that's a less problematic word if I say it's non-standard. This phosphate group could easily attach here, and or here, or it could attach in this cyclic thing here. And so going back to this diagram, this is where the analogy fails. So even though you could clearly see that probably a steering wheel or a muffler is not going to attach to a door, the problem in abiogenesis is you can have all sorts of parts assemble spontaneously in non-standard ways, and sometimes it's actually energetically. There are a lot of considerations why it would tend not to assemble in the standard ways we see, and I'll cover that a little bit more with the law of large numbers. So, so this is where some of the analogy breaks down, and this is where it gets into the idea of purified parts, which Tony Maurice talks about. If you have other substances there that can that's in the prebiotic soup, it'll make the job of these covalent bonds forming standard bonds that the bonds of life, so to speak. Maybe that's a better term. The bonds of life as we know it. It would preclude them from forming because it would gum up the system, to use James Tour's own words. I'll use the word gunk, and so I'll cover that a little bit more. One of his arguments is that he didn't have a problem with the actual experiments being conducted by the synthetic chemists. He had a problem with how they represented it. So just by way of exaggeration, this is a 747. If I showed all the factory and manufacturing processes of making a 747, that should not be problematic. What would be problematic is if I said, look at all these manufacturing processes, I've demonstrated them, these factory processes to make a 747. That proves a tornado passing through a junkyard can form it. That would be very problematic. So again, I'm exaggerating, but if one said then that some synthetic chemist came up with a process to make a ribose, and it's 90 pages long, and then he'll say this suggests how life arose. That's, from Dr. Tour's perspective and the perspective of some of us on the ID side, this is problematic because that doesn't indicate that the process is very general. It would need a highly specific, orchestrated set of events. To me, that would be more honest to say this suggests life arose out of highly specific conditions. And by the way, look how hard it was. I think that's what he was pouncing on, was that that's not right in his opinion. He thought that was dishonest, and sometimes he called it outright lies. He actually used the word lies to describe one of the papers. I would say that let me just suffice to say, I find that problematic to be extrapolating that. So I'm going to move on because the topic is mixing entropy. So even just to describe some of the process of making the jet fuel, it is an involved process to refine the petroleum to make the kerosene fuel for jets. And then even something as simple as the wires inside the jets. We need copper wires and not necessarily copper. We need metal wires and then some sort of insulation. This takes a process to make it. And so this is where, again, the purified materials come in. So these were some theological things that I said, and I'm just going to skip over that. One thing that when we're calculating probabilities, and this does actually relate to entropy, entropy can be said to be a measure of multiplicity, but the converse isn't true. Multiplicity is not necessarily a measure of thermodynamic entropy, and I'm getting way ahead of myself. But if we looked at this configuration, and these are presumably fair coins, fair quarters, 100% heads is a very unlikely configuration. We call that a low multiplicity state for these coins. That means there's only really one way we can make 100% heads. Whereas if we wanted 50% heads, there's a lot of ways we could make it, very many. And so we would say it has high multiplicity. So one way, and this will relate to the various problems in abiogenesis, where we have molecules that violate the laws of large numbers from an assembly in a prebiotic soup, and then also some other biochemical considerations. A very easy one that has some things that need to be addressed is homochirality in amino acids. We have most of life is L-amino acids, and how this can form in a prebiotic soup was the subject of Dr. Truer's discussion. He was saying some of these experiments that claim to solve the problem, he felt actually didn't solve it. And I'm just going to bypass that because some of the viewers have already seen that. Now, sugars, they're almost all right-handed. There are some like arabinose that I think are left-handed, but D-ribose is 100%. The riboses in like DNA are all 100% D-ribose. It's a low multiplicity state for ribose. And I found some really interesting things about this that make this especially difficult. So if the amino acid one seemed challenging, the one for ribose is just far worse. And I may go into that. The other thing is the linkages. And I'm going to go back to my diagram here. This is a nucleotide that we use to make DNA. The connections are always between 5' and 3', 5' and 3'. Whereas in a prebiotic chemistry, there's no requirement that it's going to hook up that way. And Dr. Tuer covered this. It could be 5' to 2', and then you can have all these strange breaks in forming the polymer because just naturally it just doesn't happen. It's kind of like flipping coins. You can't guarantee it's going to turn out one way. But in formation of RNAs and DNAs, it's 5', 3'. So this is a violation of this kind of organizational entropy. I'm not going to call it thermodynamic. Just in terms of multiplicity, this has some corresponds to thermodynamics, but I'm not going to go there right now. So this is an illustration of a DNA here, and you can see the connection 5' to 3', 5' to 3'. When we've made random soups of nucleotides and tried to connect them together, we end up getting a mix of 5' and 2'. And so again, this is a violation of the law of large numbers. And there are other things. One reason we want purified chemicals is you can end up connecting all sorts of things into the wrong places, and it's going to gum up, or I like to say gunk the system up, and you can't build this if you have the impurities. So that's when Tony Maurice was saying, let's have these pure chemicals. Why James Tour and other ID proponents consider that cheating, it's because it's going to, unless we have like enzymes that filter it out, to filter out the other chemicals in the soup, like say in the cell, the polymerases help make sure the right chemical gets there to create the bond. So in a prebiotic soup, we don't expect that we have things like those sorts of enzyme complexes, like a polymerase. Go on, Christopher.
One interesting thing about that is that if you have mixed linkage molecules, you know, a combination of 2 and 5 versus 3 and 5, you won't form stable sense antisense groups, right? So because that base pairing is a stabilizing factor, the strands that have mixed linkage are going to be degraded at a much higher rate than the ones that don't.
Yes.
And as they are degraded, once you have your free nucleotides and nucleosides, you have on each of those a chance of moving that phosphate to a different position, right? And so because only the 3' 5' are going to be stable in base paired complexes, that's going to maintain that at a higher percentage than the 2 to 5. And then those 2 to 5s are going to be more likely to have the phosphate move to a different position, at which point, if it's moved to the 3, then, well, now it's more stable, so
well,
just a thought.
I appreciate the feedback. The issue is what happens in a prebiotic context.
That is a prebiotic context.
In the prebiotic, okay, because when they had these, the polymers were having a percentage of them being 5' 2'. It didn't spontaneously go all the way to 5' 3' through the whole strand. And Dr.
Yeah, I agree with that. What I'm saying is that when you're doing a single reaction, right? So the thing that you have to remember is that you've got cyclical things here happening, right? You've got heat sources that are, you know, fairly have a tendency to degrade your compositions, right? So you've got higher stability from base pairing, and you don't get base pairing with mixed linkage. So it's true that if you just toss this stuff together, what's gonna happen? You're gonna form these molecules, and some of them are gonna be 5 to 3, some of them are gonna be 5 to 2. That's true, right? The ones that are 5 to 3 can base pair. The ones that are 5 to 2 can't. The more, and when I say that, what I mean is that the more 5 to 3 you have in the molecule as a percent, the greater the degree of base pairing you'll have with other molecules, right? So if you have a range of percentages produced, because it's random, right? And so you'll have a normal distribution of percent compositions, with, you know, most of them being about 50-50. Actually, it's not even 50-50 because I think the 5-3 is more stable anyway. But don't quote me on that.
I thought the figure was like 66% will be 5-3.
That seems right. Yeah. So it'll be, the bell curve will be around the 2/3 mark. But the ones that are shifted over a bit, as you cycle this through, the ones that are shifted over more are going to be more stable. So they're going to break apart less. And so the ones that have more 5-2 in them are going to break apart more, and now you have more nucleotides have a greater chance, or the ones that have more 5-3 are less likely to be recycled, right? And so that's what allows you to get this increasing level of 5-3 linkage, because it's more stable as a base pair. And if you're not considering base pairing, which, as I recollect, the paper that Tor was talking about was not considering base pairing, right?
Mhm.
If you're not considering base pairing, you're not going to see this effect. If you're not considering cycling, you're not going to see this effect.
Well, to my knowledge, that didn't happen in the experiment at all, because they didn't get a lot of. I mean, if we just threw this as a soup, the result of the experiment is we weren't getting consistent 5-3 of any substantial length. I mean, it wasn't a spontaneous.
You'll get some base pairing at 2/3. It just won't be much.
Yeah. So I think you put out a valid point, and this is why it's good to have discussions like this. We need to revisit this. So can I just file that away and move on? And
yeah, sure. I just wanted to raise it as a thought.
Yeah, I think, yeah. So I will point out though, for RNA, it's going to be much more difficult. And I thought he had a good point. We ended up getting a lot of these cyclic phosphate groups here, and it would actually break the polymer. And that's one of the reactions that degrade RNAs. That's why they have to be kept at such low temperatures. And so I thought for the RNA argument, he made a very good case there. So I'm going to move on, but I thank you for your point there. So just in terms of probability, low multiplicity states are more improbable than high multiplicity states as a matter of principle. That's why this pattern stands out. That's also why homochirality and consistent linkage stands out in life. And as I was if I had a picture of RNA, this is much more pronounced with the problem of RNA, because it's very delicate. It's very easy for the RNA polymer to just be blown apart. He was pointing out that a lot of places will recommend you store the RNA at minus 112 degrees Fahrenheit. At room temperatures, it will not last long. The half-life is very, very short. And these are some of the chemical things. So the question is, how do these things arrive? If they're going to invoke the RNA world as a start, there's already a problem there because of the problems of creating these consistent linkages with RNAs, which is much more difficult than it would be for a DNA. I mean, creating the consistent linkage and preserving it becomes a serious problem for RNAs because they are very delicate. So let me keep moving here. There is a discussion of multiplicity in this website called Hyperphysics, and I'll just skip that. And at some point I'll have this. Okay, so now this is the really nerdy stuff. This is to answer specifically Ben Rex's question, and this is good practice for me because I may make a separate video on this because it deserves a separate topic.
So just really quickly, I had to pull up my Wolfenden 2010 half-life of the phosphodiester for RNA at room temperature is 110 years.
What causes the breakdown of RNA at room temperature?
It's mostly that internal attack.
The internal attack being
from the 2' hydroxyl attacking the phosphodiester.
Okay, but you said
that's mostly the reason. 110 years half-life at
for a single reaction. But if you have an RNA of any length, the likelihood then that the entire RNA might be severed at one point then increases.
I certainly agree. If we're talking about any degradation, which is, of course, as you say, why it's a bad idea to keep your RNA that you're using for your study, that you paid massive amounts of money to have specifically synthesized for you. You want to keep that in the ultra cold, right? In the -80.
No, but by the way, thank you for that data point. 110 years for the phosphodiester
Yeah,
bond.
it's
Okay, thank you.
16 days at 92 C, and this is abiotic. Obviously, if you have any RNase in there, it's gone.
Thank you for the data point. See, now I learned something. This is great exchange. I wanted to say this is I had to study this when I was studying statistical mechanics. When we were learning what microstate means, I was trying to describe the coins or dice in terms of microstates. The meaning of microstates in thermodynamics is different. You can still use multiplicity, but the meaning of the microstate is different. And you need theorems like this. It just makes it miserable. So that's why, again, I say, please, guys, don't go there too quickly. This is not an argument you want to get into, and you don't need to use it. What I've said, and this relates to mixing entropy, this is a much easier way to understand it. We have marbles here, and I'm just going to assume for the sake of argument they're all the same size and weight. They have different colors. It's very hard for them to spontaneously segregate. And we have examples of that also at the molecular level. So this is the problem of mixing, and we can even give scores to this in terms of mixing entropy. There are things that spontaneously separate, like water and oil. And then we also have ways we can separate sediments. The ways we could separate sediments spontaneously in a jar with water. But the issue is that there are some molecules it's not so easy to separate. And I'll use the word again, low multiplicity. In a basic chemistry class, we'll see, this is an example actually from a basic chemistry class of mixing entropy. We have one species of gas that's represented by blue dots or blue spheres on the left, and then red spheres on the right, a different species of gas. This is kind of the low entropy, low multiplicity state when we have this valve separating them and the valve is closed. Once we open the valve, the entropy increases. And there's actually a formula associated with the increase in entropy here. How this is derived is a little bit nasty, and it takes some rumination as to why. But the point is, this won't spontaneously unmix itself. What Brian Miller was saying is, in a closed system at equilibrium, things will. There are certain processes that won't happen, and this is one example. It's not going to spontaneously arrange itself into blue and red molecules, so to speak. If you have any number of them. Now, obviously, if you just have one blue and one red molecule, it could, but when we're talking about large numbers of them, it won't, statistically speaking. And there's an associated entropy with that. And the units for entropy are joules per Kelvin, or even just the count. You can do a logarithm of microstates, but we don't have to go there right now. This also relates, there's also a thing with homochirality. Chiral, like for amino acids, if they start out being all L amino acids, they have a strong tendency to become a mix of L and D. They will racemize, they will become a mix. And I just wanted, just for the sake of completeness, there is this book on stereochemistry that highlights this fact. And this becomes a problematic. So the problem of purifying and isolating substance out of the soup often changes mixing entropy. And what I was saying with Brian Miller will argue, well, this can't happen in a closed system. And when I was saying, well, what's the point? Because we have open systems in non-equilibrium. What you really have to argue is then the mechanism. But if you argue the probability, the mechanism, you're not really even arguing thermodynamics at that point. You're arguing the probability of creating a mechanism. Like I can give a mechanism that will separate the blue and the red molecules. You keep cooling this down until you reach the melting point. Yeah, I'm sorry, the boiling point of one of the species, and then it will liquefy, and then you could separate it. That's how they separate various species of gas. So in a non-equal, yeah, I'm sorry.
Crystallization exclusion also.
Oh, thank you. So the point is, if you have a non-equilibrium system, open system, it's solvable. So why even invoke thermodynamics? The problem is, what's the probability you can come up with a mechanism that can do this mixing and separation? So a lot of Dr. Tour's stuff was talking about the problem of spontaneous being able to unmix things and the difficulty of forming those mechanisms. And I found some examples that I can appreciate why he was focusing on that. Not to mention that things also spontaneously, once you start out with a purified substance, particularly homochiral amino acids, it won't stay that way. It has spontaneous isomerization. So where was this in? Okay, this was the nerd stuff for Ben Rex. If he's still here, are you still here, Ben Rex? He asked this. A few points. Could you go a little bit more into details? The different entropies you mentioned is something I don't really understand. So first off, entropy is not a measure of disorder. Just need to point that out. That's just a side thing. That's kind of an obsolete view, and a very respected chemist was pointing that out. That's obsolete. Don't use it. And also a forgotten creationist book endorsed by a Nobel Prize winner, Eugene Wigner. He gave a counterexample that showed that entropy can be correlated with disorder, but it's not the same thing. And so back to Ben Rex's question, and I'm sorry, this is just getting really technical, and it's understandable the viewers are dropping out because this is far removed from Dr. Behe's talk. From this book, this was the founding book of intelligent design, Mystery of Life's Origin, 1984. You can get it for free on the internet. There are PDF copies available for free. I think that was, you can buy them, but they made the book available for free. It is a classic. And here it explains entropy in terms of configurational and thermal entropy. And the configurational entropy is that can be things like mixing entropy. The physicists will totally cringe at this separation, but a lot of materials engineers will use this idea of thermal and configurational entropy. And we actually calculate it. It's easier to talk about changes rather than absolutes. So let me see if I could find. When I give a separate lecture on this, I'll cover the Sacro-Tetrode equation. You could actually see it when I'll provide a spreadsheet. You could put in your own numbers for changing. Like if you change the volume, you change the configurational entropy. If you increase the temperature, you change the thermal entropy. And so you could change these terms in the Sacro-Tetrode equation for ideal monoatomic gases. You change one term here, you change the thermal entropy, you change the volume, you change the configurational entropy. And so how does this relate? What's actually happening is your blue molecules here, when we open the valve, we're making it actually bigger for the blue molecules to occupy that space. And so we change the configurational entropy. And likewise for the red, when we open the valve, the red occupies more space. And this is actually what you're seeing here in these terms, the change of entropy based on just changing space. So that's the difference in configurational thermal entropy. And it actually, when you combine it, configurational with thermal entropy, you get the total thermodynamic entropy. So that is the difference And so that's the big can of worms that got open when I even mentioned the term configurational entropy or mixing entropy. What's the differences? To understand why it all gets put together that way, it goes back to this Liouville theorem, and that's not anywhere we really need to go, so to speak, to discuss abiogenesis. But that was because we had a viewer question on this, and I can cover that more, Ben Rex, if you want to be tortured with the details, or if anyone else wants to be tortured with the details, because that's what I like to, that's what I spent. That was one of my more enjoyable graduate classes, was be considering these questions. And that's really all I had. So there you have it. He said, I don't mind this kind of torture. So with that, I did have, let me see if I have an example. If I had a picture of the ribose that I wanted to discuss with you.
While you're looking, let me just go ahead and strongly agree with you that I always get immediately concerned whenever I see somebody about to use entropy as an argument, no matter what they're trying to argue for, because entropy is not the simple concept that people who aren't in the sciences think it is. Like, people who are not, and thank you, by the way, for pointing out that entropy is not simply order and disorder. It's sort of a pet peeve of mine when I hear people say that, entropy is just disorder. Like, stop that. It's painful.
Yeah. And I talked to a biochemistry author. Larry Moran he runs a Sandwalk blog, and he's also a professor of biochemistry, and he has a biochemistry textbook. And I asked him, I said, you said it's order and disorder in your textbook. He said, yeah, that's an approximation. We know it's wrong. So I had him on record publicly saying that's not correct, but he put it in there.
If you get Voigt and Voigt, they will very clearly state that entropy is not simply order and disorder.
Yeah, and just for the reader's benefit, the reason this all came about was kind of an unfortunate comments by both Boltzmann and Gibbs, who were the founders, and they were kind of passing comments. Boltzmann did use the word order or disorder, but it really wasn't in his derivations. And same for Gibbs when he used the word mix-upness. It was in an unpublished paper, I believe, or uncompleted one. And so as long as you don't try to bring the proof for it, I'm an engineer after all. Yeah, I totally get that. So that's how it kind of, unfortunately, quote unquote, evolved and became part of the nomenclature. I mean,
there are so many intuitive examples as well, and that's unhelpful. Is that there are a large number of extremely intuitive examples of entropy that can be represented, that are precisely correlated to disorder, right? But the fact that you have some examples that are one-to-one correlations doesn't mean that is actually the phenomenon in play, right? And that, I think, is a big part of the problem, is because people like to illustrate it with, here's this simple example of increasing entropy. And then, and people then think, oh, well, now I know what entropy is. Well, no, you know an easy example of entropy.
Well, I'll give correlation is not the same identity. We can correlate reading ability with shoe size, just because the little kids, as they grow, they're also, their shoe size increases. So does their reading ability. It doesn't mean shoe size is the same thing as reading ability. And that's the example I talk about, with entropy. You can have
great analogy.
Yeah. So now this is one I was talking about the problem of purification. For those who know, think it's this beta. Correct me if I'm wrong here, CRISPR. This molecule here is the D2-deoxyribose, and it's the beta form where you have the OH group pointing up. This is what they would call the furanose molecule. Now, in solution, if you put ribose here, you actually get 5 isomers. What ends up happening is when it dries, you only get the pyranose. You only get the pyranose. Let me see if I had to look this up. When it dries, when it crystallizes, you only get the pyranose form. And
for the
I have,
if you can go back a slide.
yes.
For the non-chemists among us, that's the thing at the bottom where there's not the overhang.
Right. This is the pyranose forms here. So where am I leading with this? We're talking about purity. For the DNA, you could see we have this very specific, this is the molecule that's in question, this ribose. All of them, all the riboses are in this beta furanose form. But if we took the ribose molecule, if we even took this furanose and threw it in water, we would get these 5 forms here. And that's a legitimate problem of, so they've had to work around this problem. I don't know that they've solved it in the synthesis, because if you end up having a mix like this, how can you form only one class of nucleotides? I'm not saying it's impossible, and neither does James Tour, but at a certain point it starts to become, you start to see all these problems develop, that even when you start with a purified chemical and you just throw it in water, you're going to get 4 other things that you don't want. And sorry, I saw you threw something in private chat, and like I said, I don't read private chat.
Yeah, that was just I posted it to the extent I could in the outside chat as well. But I had referenced the 2011 review article by Woofenden, which
so I just wanted
several times now. But if you wanted it, it's there.
Okay, I posted it also in the public chat. So I just apologize to you. The only reason I mentioned it is I usually don't read the private chat because I'm operating even when I'm on someone else's.
I understand. I figured that you might see it later, and then because I referenced it, so I wanted to make sure that it was available.
So people on my side of the argument will point to this one of these. This is one of the problems with the formation of of consistently the same nucleotide, because if one wants to form not just consistent linkages, but even having the same nucleotide or whatever, where you have the same kind of ribose here always, the purification problem becomes a challenge. So they do have dry synthesis, but the problem with dry synthesis is that this form of the ribose, I'm sorry, totally blew that. This form of the ribose happens only aqueously. The dry form is always in the pyranose form. And if this is the wrong citation, I have another one, because that was, here it is. In the solid state, only alpha and beta pyranose is found. Okay, so it can occur as pyranose or furanose, but in the solid state, only alpha and beta pyranose is found. So when we dry this aqueous solution that has these 5 parts, you don't get this up here, and it's really only this one we want. So I found this really interesting. They do a lot of dry synthesis with oligonucleotide synthesis, but what they have to do, they get around that by having a preloaded, it's called DMTO. I think they derived it from a pre-existing living organism, and they're able to get around it because it's a full nucleotide. It's not just the ribose. So trying to form just the ribose and then trying to connect it to a nucleotide becomes challenging. You can't use dry synthesis, And in an aqueous environment, you naturally get all this junk. And so James Tour was pointing out that you can't just synthesize this and say, oh, we've solved the problem, and then go buy the purified, like say a nucleotide, because he's saying this was skipping steps. It's not exactly right to represent it as saying this is a legitimate pathway. And so this is, that's one example. And so this is kind of like an example of impurity and mixing. And I won't even use the word entropy here to just mess things up even more, because this is one of the things I looked into. So where did I get some of this? I didn't get it on my own. There is a book. Let's see if I have it here. I'm gonna have to just Google it. CRISPR, if you had any thoughts before I close the stream on this, I'm just going to show the book where I got some of this information. And some of it I came up with on my own, but the inspiration from it was Change Tan's book.
No, I mean, not really, nothing specific. I really appreciate the opportunity for dialogue. There's I don't know of a single person who would deny that abiogenesis is a complicated topic, right? Like, I don't know of anybody who would suggest that it's a solved. Well, I've heard people say it's a solved issue, but I don't think that they are at all studied on the subject matter, so let's leave them aside. People who are actually qualified to discuss it, I don't know anybody who would say it's a solved problem. On the other hand, I think that progress has been made, and I think that denying that progress has been made in the field of abiogenesis would also be silly, which I haven't heard you do here, just to be clear. So yes, there are obviously problems, big problems in some cases, but also progress has been made. So where's it going to end up? It's hard to say.
Just to clarify my own personal thoughts, I actually don't think that what is advertised as progress is actual progress. And one of them I was highlighting, it's like all these synthetic experiments being represented as progress towards solving how it's done in a prebiotic soup. I have problems with that. I don't have a problem saying that if we said that these synthetic experiments show how difficult the process, how unlikely it would have to be, I'm a little bit more fine with that.
So it's progress in the sense that it's progress in the sense that I've heard people and seen people suggest that that certain pathways are or should be impossible abiotically, right? And so when you find an abiotic pathway to achieve some end product, right? That is progress over somebody saying it's impossible to achieve that end product abiotically, right?
And
I think that that's progress.
Yes, in the sense that it had to be an orchestrated set of experiment, a laboratory, very carefully orchestrated, very intelligently designed and orchestrated. And I'm
so I agree with that.
If they would report it as that, I would have less heartburn.
But consider this. Consider this. Let's say I think I know a pathway that could have produced an abiotic pathway that could have produced final product X, right? And let's say I do an experiment of the sort that it seems like a lot of people on nominally your side kind of want, and I just willy-nilly throw things together and see what happens, and then I don't get X. Does that mean that my pathway doesn't work? Or does it mean that I don't have a sufficient sample size? Or does it mean that I haven't considered who knows what factor, right? Because the point is, because if I do it under careful control and it fails, right? Then we can say, hey, that pathway's out. Can't work at all. And if I do it under careful control and it works, then we have a second question, right? Now we can move on to the second question, which is, how do we control for yields? How do we do various selectivity processes that can maintain this form over some other form? How can we do X, Y, Z, and so forth, right? Because at least here is a pathway that works. But if you don't control for it at the start, you can never say that the pathway works or not at all. You can't even say that the pathway doesn't work, because you have somebody else say, "Well, you didn't consider this." And so I find this. I understand where that criticism is coming from, but also I take serious issue with it, because, I mean, you got to show that the thing works first, right?
Well, I wanted to say the pathway obviously exists. It's called a living cell.
Well, that's a living cell is not an abiotic pathway now, is it?
Well, see, that's the issue. Is these, when we say it's an abiotic pathway, it's an abiotic pathway plus an intelligently designed, carefully orchestrated sequence of events. That's not what you would find in a prebiotic scenario where we have lots of random events. The more careful steps that need to be in that orchestration, the more things that can go wrong, which is the higher probability of that going wrong. And that's really the point, is, well, at what point do we have so many careful processes to make even a minimal life, where even just a few things going wrong and the probability of a few things going wrong is astronomically high from deviating from that? At what point do we start to say that we can legitimately say this happened naturally? I mean, there's a point where for some of us, it's just too hard to believe. That's so where, again, yeah.
I understand where that's coming from, right? But my point is, if we do the experiment the way you're suggesting we do the experiment, right, which is no careful control, then
no, I'm not saying we don't do the experiment with no careful control. I'm saying we do the experiment, but you can't represent this as being something that would happen in a prebiotic environment. You can't represent it as being a high probability scenario.
What? So I've read a large number of these sorts of papers, and the authors, from what I've seen, are very quick to comment that there remains issues with selectivity and purity and yield, right? But
and I appreciate that.
But that's not the point of the experiment that they're doing, though, right? Because before you can talk about how do I increase the yield of this reaction pathway, I have to show that the yield exists, right? Because if the yield is 0, I can't increase the yield, right? Because there's none, right? You can't do anything. The pathway doesn't work, right? If the yield is 1%, then you can talk about how do you increase the percent yield? How do you increase the purity, right? How do you increase the selectivity? How do you do whatever? But if the yield is 0, there's nothing to be done.
But I just have to say that if the process to make even that beginning yield had to be carefully orchestrated, I think it'd be a lot more, I think, less misleading to say we wouldn't expect anything as carefully orchestrated as this to be present in a prebiotic Earth. I think that would be honest.
Well, but the thing is, I don't know the extent to which it does have to be that carefully orchestrated. The fact that the researchers, in setting up their experiment, are extremely careful because they want to get a result that, once they have the result, is conclusive, right? And so the issue is that they go through all of this effort so that at the end of it, no matter what the result is, nobody can say, well, if only you had done X, then, right, They don't want that. And so it could be that if you got rid of, 5 of the steps in the middle of their protocol, they still would have gotten a yield. But we don't know, because they had to be, they were being careful for the purposes of peer review, not necessarily for the purposes of chemical necessity.
Well, I'd point out, this is, if they were being, I think, more complete and less biased in their approach, I think a more accurate picture would be, if we left this step out, this is what would happen, and they're not reporting that. And I think that's important, because they if we want to have a balanced picture of the difficulty, if they're signing their name and saying this suggests a possible pathway, I think it's reasonable to ask, how probable is this possible pathway? One can actually give clarity to that by reporting, like if we didn't do this step, this synthesis would not come about. And if we have, like in one example that Dr. Tur pointed out, was 90 pages of the recipe, and you snip one page out, I think that would be a more balanced thing. I wanted to highlight something Mr. Jetty said, Mr. Jetty 89. Dr. Tur said this, he was saying that if he had students that approached him, or thinking about becoming scientists, presumably these would be his PhD grad students. If they approached him and said, what lab should I join? Or let's say these are his undergrads and they're looking for a PhD program, or his PhD students looking for a postdoctoral fellowship. If they approached him and said, what field should I study in chemistry? He said, don't go to origin of life. And he said this in the last episode, because your professor will die before he figures out the origin of life, and you will die before you figure it out, and your students will die. I just wanted to highlight why to the viewers what that meant, and why I put it there. So thank you, Mr. Jetty.
What's the line about?
I didn't mean to break your train of thought there, Christopher. I think
I
appreciated hearing from you.
I both think that I had finished that train of thought, and also that it raises an interesting note, which is, have you ever heard the comments about wisdom? It's the wise man who plants a tree he knows he'll never sit in the shade of. I've got that quote wrong, but maybe you've heard it.
No, I've not heard it.
Yeah. Hopefully the point there is clear.
I think I sort of get it. So for people on my side, it's like, and I think Paul Nelson echoed this sentiment. If life was the result of a miraculous process, he'll use the word intelligent design. I'm just going to stretch that a little bit from his original phrase and say a miraculous process. And certainly a miraculous process we cannot ever hope to repeat in the lab. Let's just say hypothetically that's how origins actually played out. What that means then is that all these attempts at naturalistic experiments that demonstrated if life is designed that well to resist naturalistic explanations, it's going to fail permanently. And so that was the sentiment of, your professor will die, you'll die, your students will die before they figure it out. Because as a matter of principle, if on the premise that it was miraculously created and by a process that we'll never be able to do ourselves, it'll never be solved. And so there is kind of this, there are two directions people can go. They can, maybe three. One direction is they'll accept a miracle and intelligent design. The other direction is pure naturalism. And then maybe the third is like, I don't care. And that probably defines the divide right now, is that the ID camp will say that the origin of life experiments in terms of trying to show it can happen naturally will consistently fail. There'll be problems with the with arguing that this can happen naturally. So with these very carefully orchestrated experiments, probably a little bit more robust reporting and saying, okay, this is the sensitivity of the process if we remove these steps. And if there are a lot of them that you can remove, there are a lot of steps and a lot of them are critical enough that if you remove them, the end product will not appear. That'd be pretty important. And that's a lot of what Dr. Tour argued. So for a lot of us on the ID side, we thought he made a very good case that they haven't actually made progress. And he actually did finally in his final episode, made the case that he doesn't believe that there's going to be any more progress.
I mean, that's a bold stance to take.
Yes,
I don't know. I don't know that I'd ever want to argue against no progress will be made. Argue against scientific progress, because even if you're right that it is on a fundamental level unsolvable, right? That doesn't mean that we can't approach an answer asymptotically, right? At which point we'll still make progress, and we'll make progress all the time. We'll just never get there.
I do want to say this. Oh, go on. I interrupted you.
To close that thought, arguing that no more progress will be made. Ooh, that's a tough one. I don't think I don't think I would ever stake out that position.
Yeah, but what I think, and I'm glad you challenged me on that. I'm going to clarify a little bit. What Dr. Tour was saying, for every step we may think that we've solved, we find, just historically speaking, from what we've seen, we end up finding way more problems than we think we've solved. So going back historically to, like, say, the Urey-Miller experiment, and I was in junior high and they were teaching that in the mid 70s. There was a lot of optimism that this was just a simple chemical reaction. We didn't need very specific conditions to start life. Now that we've moved forward and we're seeing these other chemical issues, there are people even now. I mean, what happened is we were very close to solving life. So that was in the 50s. And that was repeated even in my 1975 classes that Urey-Miller was suggesting we're close to life. But even around that time, they began floating the idea of panspermia because they were realizing the difficulty. And now it's gotten to the point we have someone like Koonin who said the odds of even the RNA world of creating a transcription translation system would require a multiverse. So we've gone from maybe simple chemicals, then to outer space origins, now to multiple worlds. The trend has not been, the opinion trend has not been kind of seeing that this is going to be easier. So to your point, I think maybe I will use a better choice of words in saying that there won't be progress. And so I thank you for pointing that out. Maybe I need to think on what Dr. Tour was saying, is that he expects there are going to be the number of problems that will emerge will outpace whatever little we learn through our synthesis experiments. And I'm glad synthesis experiments are being done. They are helping clarify the issue for both sides. Of course, we'll read those synthesis experiments and say, see just how difficult it is to make life. And other people will view it as progress and showing that life can emerge naturally. But I think I'm not saying, although some ID advocates have said, let's shut down funding of this, I'm ambivalent to that. I think learning anything about synthetic chemistry will be beneficial, just in general.
That sounds reasonable. Yeah. I think that would be a reasonable interpretation, possibly, of what he might mean. And that would be a framing that I think I could, it's not one I would make myself, but I could understand it.
Yeah, and that would be kind of in a sad way. That's a testable prediction. I mean,
We'll see.
And I mean, I've had one of the professors I mentioned in this talk, James Treffel. He and his colleague Robert Hazen at my undergrad alma mater in 2005 said, we're going to solve the origin of life problem in 20 years. And I told him, I said, I'll buy you a beer if you do. And so that's 2005. So there's 4 years more if he wants to collect my beer bet. So I don't have anything more. I'd like to, I think I'm going to shut the stream down. And I'd like to thank CRISPR for joining, Dr. Dan for starting this whole afternoon, and all the people that joined the stream. Forgive me if I forget some of the names. So that would be Emery and Erica and David Neff, and CRISPR and Dan have joined, and Otangelo joined earlier. He was the first to join. I'd like to thank all the viewers that have joined us in this fun, and who also watched Dr. Behe. So it's been great. And CRISPR, I'll let you have the last word before I just give some final administrative things.
No, just thank you so much for having me on, and the fun discussion of chemistry. Not the field I usually talk about, but I'm at least passingly familiar. Hopefully, I was clear that I've looked into this stuff before. It's not my usual area, but I'm comfortable enough talking about it. And hopefully, we can do it again sometime.
Yes, that sounds great. You really added to the discussion here. And Ben Rex, it's been fun. And I will try to make a video just for you for this mixing entropy stuff, because this was totally on the fly from a draft. And I'd like to thank all the viewers, and take care and have a nice day, and God bless you.